Recording Shakir live nahi. Tell me when to start.
On a count of three.
3, 2, 1.
Shall I start? Okay.
Yes, sir.
Okay. Good evening, everyone. I'm Gaurav Kant, and welcome to the Andrology workshop of ACE. On behalf of Dr. Ved Prakash, President of ACE, I welcome you all. So we have very esteemed faculty with us. We have Dr. Prabhakar Singh, me. We have Dr. Kon Mitra, Dr. S.S. Vasan, Dr. Jindal. Vakil is here. We have Dr. Sejal Gupta from USA and Gloria Martinez. So I just want all of you to be there, stick to the seats. It's going to be very important for all of you to improve your practice in the IVF and andrology. So without wasting time, Himanshu, can we play the inauguration video?
शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः.
Thank you, Himanshu.
Hello.
Hello, Ratnadi.
Good afternoon. Today is the first day or day of workshop in 9th Annual Conference of Academy of Clinical Embryologists 2021. I would like to convey our heartfelt thanks and gratitude to ACE Central Committee for giving us the privilege of hosting ACE 9 Annual Conference this year in Bengal on behalf of all people in Bengal related to assisted reproductive technique practice. I welcome all the delegates, faculties, and others who are working behind the limelight for the success of today's program and the main conference. You know, in the midst of enormous suffering, inconvenience, and loss, at last we all are over here today because of the continuous support and cooperation from a Central Committee member, and of course because of the team spirit, hard working, and dedication of local team member. We know that workshop is the main attraction to an embryologist. Hands-on training is utmost important to an embryologist rather than theoretical discussion only. It is partially true for the clinicians also. Hands-on training on ultrasonography, oocyte retrieval, and embryo transfer are essential part of their day-to-day work in an ART clinic. I am confident that the knowledge and the experience of the faculties in today's program will take the workshop program to a different height. I am sure that you all will enjoy and gather practical experience from the workshop program that might help you in your day-to-day work later on in the lab and in the OT. I hope that you all will have a great time together. Now I am requesting the hall coordinator to start the evening session. There are two workshops in embryology, one on andrology from basic to advanced, and the other on uh, standard operative procedures and the instruments of your lab. Best wishes and thanks to all of you.
Thank you, madam, for your lovely message. We're definitely going to rock the conference and the meeting. So we have the first speaker with us, Dr. Prabhakar Singh. Can we have the CV, please? So Dr. Prabhakar is a director at Nimaya Women's Center for Health and 21st Century IVF Center, Surat. He is the MBBS and MD, and he has completed his MSc in clinical embryology from Leeds. He has a vast experience of 10 years, and he's also the head of embryology training offered by 21st Century Hospital. He is an avid reader, book lover, a marathon runner, and a very nice human being. Welcome to you, Dr. Prabhakar Singh.
Thank you, Gaurav, for that.
Good evening, friends. First of all, I'd like to welcome all of you to ACE 2021. And I'm thankful to the organizing secretaries and the organizing chairperson, especially Saroj and Dr. Gaurav, for inviting me to give this important talk. WHO guidelines for semen analysis have always been an awaited event in our calendar. And this year, too, after almost 11 years of using the old guidelines, we have all been waiting desperately for the new guidelines to come up. The launch of the sixth edition in August was one of the biggest web-based event in this year in terms of human reproduction. Today, I'll just go through a little bit of what the sixth edition is taking forward. I wouldn't say it is different from the fifth edition, but it is an evolution, it is a progress, and it is a movement towards the next step rather than changing the guidelines completely. I think the committee has done a fantastic job of evolving it and making it more relevant in terms of our current times. Now, the whole point of doing a semen examination is a part of the male infertility workup. So when you want to try and see whether there's what are the possibilities that are available for the couple for conception, whether they can conceive naturally, whether they will need some sort of IUI treatment or natural cycles, or they will end up in IVF or ICSI, the most critical factor that defines this is the male workup. The male workup is will identify the following conditions: a potentially correctable condition, because that is what we want. If we have a condition which we can correct easily, then there's a lot of stress and lot of process which is removed. Irreversible conditions suitable for ART. Now there are many conditions which are like severe azoospermia, CFTR, which are irreversible. Conditions in which donor insemination or adoption are the main options, and any genetic abnormalities that may affect the health of the patient or offspring, especially if ART is used. Why is it difficult to make reference values? The problem with semen analysis is that the spectrum of values that we get is widely different in different studies. It is almost impossible to get a representative sample of the whole population in which we can define a cutoff whether this patient will conceive naturally or this patient will not conceive naturally. And that is what the challenge has always been while coming out with these guidelines, with studies based out of say 3000, 8000, 6000 males in different groups of subfertility, recent fathers in whom we have done semen analysis. We always get a very wide variation, and that has been the real challenge in the guidelines. To circumvent this problem of having reference values, the guideline did a very interesting take on this, and that is they came up not with reference values, they've come up with decision limits. Now these decision, eventually any report or any value that we get from the husband or the male partner or from the male from the semen examination, we use it to take decisions whether we need to treat this condition, whether this patient will need IUI, whether this patient will need ICSI. And that is what the decision limits have been identified for abnormal ejaculates. Then there is the change in the motility grading system. We have gone back to the fourth edition where there used to be 4 criteria for motility. Then methods to detect sperm aneuploidy and sperm genetics and DNA fragmentation are now part of routine examination, and they are highly recommended. ROS testing has been accepted as an important parameter, so that has been added, and there is an added chapters on sperm preparation and cryopreservation. Now, decision limits. The decision limit, says the manual, is based on clinical and statistical considerations that point to a need for a certain diagnostic or therapeutic intervention. Introduction of the concept thus seems to underscore semen examination not to define a man as fertile or infertile or subfertile, but rather to decide next steps in terms of further evaluation and treatment. So, reference values for a specified population are not and never will be clear delineation of fertile and infertile men. So, hence, this very important change in the way we report our values and in the way we use these values to do next. So basic examination remains the same. Factors influencing the ejaculate like size of the testicles, endocrine status, illness, medications. An additional microscopic evaluation has been added is the odor. Now this was lacking in the previous one, and that has been added in the current guideline. Microscopic evaluation, wet preparation needs to be made. There is a huge chapter on how to do that, how not to do that. So all the technicalities is currently, means I will not go in details in that. And you will need to count at least 200 spermatozoa. So this is very nicely given, it is very detailed, but it has moved to the appendix. So the major guidelines don't have too many values, they have actually moved to the appendix. So sperm count has also been changed into a new way, and as we discussed that the decision limits have been introduced rather than strict reference values. So normal sperm count is now more than 20 million is considered normal. A count of 10 to 20 million is considered a borderline count, whereas whether to consider it as a count where you want to give some treatment or the next step of planning that you need to do will depend on other factors like motility and morphology. A count of 10 million with say maybe good motility and morphology, you might still want to try IUI or even natural planning rather than going for ICSI. And a count of say 20 million with say very poor motility and morphology, you might want to go in for a little bit more aggressive treatment. Similarly, motility has been categorized as normal as more than 50%, borderline 35 to 49, and pathological less than 35%. So again, there is a 3 levels of decisions that need to be taken. The sensitivity, it is important to know that absence of spermatozoa from the aliquot examined does not necessarily mean there are absent. Sometimes it happens that we end up doing a semen analysis and we don't find even a single sperm, and this particular sperm, then this sample we report as azoospermia, but we need to do lot more tests to actually say that there is not a single sperm. Morphology, again, more than 14% normal, 4 to 14, 13 is borderline, and pathological is below 4%. A normal morphology does not mean that the sperm cannot carry another cause of pathology. An excess residual cytoplasm, usually located behind the neck and around the midpiece, is usually stained green if colored red. It indicates abnormality. Morphological defects have been associated with increased DNA fragmentation, as we all are aware, and increased incidence of structural and chromosomal aberrations, immature chromatin, and aneuploidy. Hence, we need to classify the head, neck, and tail, and the added excessive residual cytoplasm. Now, extended examination of DNA like sperm DNA fragmentation and genetic and genomic tests, this has been added into the 6th edition, and there's a huge, a pretty big chapter on how you need to perform DNA fragmentation, the different methods, the pros and cons. What I would like to highlight is that now the halo has also been categorized into big halo and small halo. So what we usually see is we usually report as with halo, without halo, and degraded sperms. So now the halo has also be large halo, medium halo, small halo, without halo, and without halo, which are degraded sperm. So this is how the category of a DFI sample needs to be reported, which is slightly different from the previous rating. Now, advanced examination ROS testing is a physiological phenomenon. We all know what the role of ROS, which plays in the semen examination. DNA fragmentation can be caused by oxidative stress, and the detection of excessive amounts is an important criteria for taking decisions. Sperm preparation techniques, there's a chapter on MAX, magnetic activity cell sorting, though the guideline does say that it is not the Cochrane database review is has not shown great value in doing this, but there is a chapter, and it just shows that sperm selection techniques like microfluidics or MAX will have a role in future as there is more data coming in, and as we get more studies published. Cryopreservation vitrification has been added in the guideline, though the guideline does say that vitrification is still for sperm samples is still in infancy, and there is a long way to go before we can actually use it for our patients on a day-to-day basis. But reports have now started coming in of freezing micro-TESI samples, TISA samples in devices or in say these droplet sort of vitrification, and that will it will only improve. Vitrification of sperms is here to stay, especially as male infertility and azoospermia cases have started becoming on the rise. To conclude, the 6th edition refrains from giving reference value for normal and abnormal, and it rather takes the view that decision limits are a better, wholesome, and more is a better way of evaluating the semen sample rather than going to strict criterias. The motility criteria has reverted back to the 4th edition of rapidly progressive, slow progressive, non-progressive, and immotile. It introduces the use of expanded tests like DFI, ROS assay, genomics, and molecular tests to expand the array of tests which are recommended for most patients. And it introduces sperm vitrification as a suggested method and use of MAX in sperm processing techniques. Thank you so much, and I'll be happy to answer.
Thank you, Dr. Prabhakar, for the wonderful recorded talk. So Gaurav, we are taking the questions after the session, after the first session, or you want me to request?
You can wait.
Let me take the questions after the session. Yeah, sure.
And Dr. Bhakar, we are waiting for some more questions. Then we'll take after 2-3 talks, right?
No problem. I'll log in again.
Thank you. Yeah. So I request the tech team. Yeah. So the next talk is by Dr. Gaurav Kant. He's working as a senior embryologist at the Kansa IVF Center. So welcome, Dr. Gaurav. So Dr. Gaurav is co-convener for Special Interest Group Andrology, Indian Fertility Society 2022. He was co-convener for SIG Embryology IFS 2018 to 20. He was one of India's first embryologists to be certified with both prestigious international societies, American Society for Reproductive Medicine in 2016 and European Society of Human Reproduction and Embryology in 2017 as a senior embryologist, and in 2016 as a clinical embryologist. He was awarded with a SAR Champion Award in 2019 and Best SIG Award with IFS in 2019. He has around 11 international presentations in ASRM, ESHRE, and IFS. He did his blastocyst biopsy and cryoposition training from Barcelona, and he's one of the keen young researcher in the field of andrology as well as in embryology. So I welcome Dr. Gaurav, and his talk will be on sperm morphology. Over to you, Dr. Gaurav.
Thank you so much, Vakil, for the nice introduction. So, hello everyone. I'm going to talk about something which is very relevant. That is the sperm morphology, the most controversial parameter. So, I want to start with this picture.
Sorry to interrupt you, Gaurav. We are not able to see your screen.
You are not able to see my screen. I'll do it again.
Yeah, now it's visible.
Okay, no issues. Thank you so much, Vakil.
Okay, thank you.
So I'm going to speak about sperm morphology. It is the most controversial parameter, I agree. So I want to start with this picture. This picture is drawn by Leeuwenhoek because he has the first microscope and he was the first person to see the sperm as well. So this is the sperm he has seen of rabbit and dogs. Even here, I cannot see a single normal sperm. So calling something normal is very ambiguous itself. For me, what is normal is which is not affected by disease, which carries a normal or intact DNA. But looking at the normal sperm, can we say that it is intact? It has intact DNA and the genetic content is normal, or it is not apoptotic? It is difficult to say that. Let's start with how we know what is normal, how it all started. So there was no ray of light from the God which says that this particular sperm is actually normal. So this is all the evolution. This all is the hard work of scientists, where the people worked hard to find out what actually is and how actually the normal sperm looks. So this all started from the study of Freund in 1966, and he took a very basic concept. He says, whosoever is fertile, take the semen sample of them, and whatever is the morphology should be the normal morphology. But he was not able to convince people. The prognostic value of those sperm parameter was not that great. So later on, this Tyberg strict criteria, Kruger strict criteria came in, where the people take the sperm from the post-coital spermatozoa from the internal cervical os. And this is how the sperm looks. This is how the normal sperm looks from the PCTs. And they were able to correlate more with the fertility and the sperm parameter now. So how a normal sperm look? This was given by Ellison in 1971, which says that a normal sperm should have a smooth oval configuration with well-defined acrosome constitute around 40 to 70% of the head. The normal head length is between 4 to 5 micron. The width is between 2.5 to 3.5 micron. The head width must be between the 3/5 and 2/3 of the head length. There should be no neck midpiece or tail defect. The midpiece should be around 1 micron and the length should be around 1.5 times of the head size. And the tail should be around 45 to 54 micron. This was given by Ellison in 1971. So now we know how a normal sperm looks. We know the dimensions. This has been diagnosed now. So can we do something? Can we find out that if the sperm is abnormal, can we say that if the sperm patient having 0% morphology, normal morphology, can we predict that this patient is not going to conceive naturally, or this patient is requiring IUI or IVF or ICSI? So let's look at the literature, what the literature talks about it. So the first study I want to present here was published in 2017, where they says that men with a complete absence of normal sperm morphology exhibits high rate of success without ART. So in the study, they took the patient with 0% morphology. All sperm were abnormal. And they try to find out whether these kind of patients need ART or not. So they took the two groups. In one group, there was 0% morphology. In second group, they have more than 4% morphology. The study was conducted from 2010 to 2013. Follow-up was there for 2.5 years. And they find out, even with a 0% morphology, patient can conceive. There is chances of 29.2%. If you don't have a normal sperm, you still can conceive and deliver a baby. Now I want to bring your focus to the C image. See, if the patient is conceiving, 75% of patients are conceiving just natural. Only 25% requires the IVF. Now look at the image D, where they says that all patients who conceived in first attempt with 0% morphology naturally, but spontaneously, conceived again without any help of ART. So we can say that strict morphology should not be used to predict the fertilization, pregnancy, or live birth rate. In men with 0% normal sperm, alternative modalities should be considered before immediate IVF. Now let's move on to the second thing, that is the IUI, whether if you have a poor sperm, whether we need IUI or not. So there is a very beautiful systemic review and a meta-analysis published in 2017, where they see the clinical pregnancy rate after IUIs, and Kruger strict criteria was used. The author used the segments of less than 1% morphology, 1% morphology, 4%, and more than 4%. So all female were between 25 to 40 years of age, and sperm count was more than 10 million. So this is a huge data of 41,000 cycles. And at the end, they says that there is no significant difference in live birth rate or pregnancy rate when you have less than 4% morphology, more than 4% morphology, less than 1% morphology, or more than 1% morphology. There is another paper published in 2019. They have a data of 984 IUIs. They again says that there is no difference in the live birth rate when you have less than 4% or more than 4% of the normal morphology. And the babies born out of this less than 4% morphology were completely normal and comparable to the more than 4%. Now let's move on to IVF and ICSI. Let's compare them. Now we know about the spontaneous conception. It can happen. IUI doesn't require it. You should not go directly for the IUI until unless the patient, the female, there is some female subfertility. Now let's move on to IVF and ICSI. So there is a beautiful paper published in 2007, and they compared where the post-was count was more than 2 million, and they found that there was no difference in the fertilization, clinical pregnancy rate, live birth rate when you compare teratozoospermia in IVF or ICSI. There's another paper published in 2008, and they also acknowledge the same. There is no difference even in the embryo grading. The blastulation rate was same and the live birth rate was same. When you compare IVF versus ICSI in teratozoospermia, even there was a higher blastulation rate because they think the female factor was there. Now to see in sibling oocytes, we take the sibling oocytes and we did the IVF and ICSI. What is the result? So this study was published in 2012 in Assisted Reproductive Genset. In one group, they have 258 patients. The varied morphology was more than 4%. In another group, 183. And they found no difference in the grading of the embryo, pregnancy rate, and the implantation rate when you compare more than 4% or less than 4% of sperm morphology. Even the author goes ahead and try to find out into different subgroups in more than 4% morphology, 3%, 2%, 1%, and less than 1%. Even there was no difference in the fertilization rate, embryo quality, implantation rate, and pregnancy rate. If the morphology is so important, why IMC is not able to prove itself? The Cochrane database, where we all rely, says that there is no benefit of doing IMC over ICSI. So till now, we can say that, yes, when we have normal and abnormal sperm, we can conceive normal naturally. IUI is not needed. And if you have a good sample, you can proceed with IVF. ICSI is not required. But there are some other issues as well, which I want to bring here. There are studies which talks about the aneuploidy when you use the abnormal sperm. They say that there is a higher incidence of aneuploidy when you use sperm with abnormalities. There is one more study which talks about there is a higher trisomies and monosomies when you go for abnormal sperm. So why it happens that the study says there is no difference, but the aneuploidy or genetic components could be there. So the simple reason is that when you go for less spontaneous conception, or you go for IUI, or you go for IVF, there is a natural selection of the sperm by the zona pellucida, by the granular cells, by the female body when the sperm travels from inside. But when we come to ICSI, not everyone is performing ICSI nowadays. There are highly skilled people. They can choose the best sperm to inject inside the oocyte. So there is another thing which we should know, that is that teratozoospermia with monomorphic abnormalities like globozoospermia, macrozoospermia, or acephalic spermatozoa, they should be avoided while we are doing the ICSI cases. So at the end, I want to say that teda hai par mera hai. All the normal sperms, whatever are there, they belong to the patient and they can give you the pregnancy. Thank you so much.
Thank you, Gaurav. Thank you for the wonderful talk. Manshu, tech team, over to you for the next talk.
So now I want to invite a very important member of faculty from our team, Dr. Konkon Mitra. Dr. Konkon Mitra is he did her MBBS from JNMC Belgaum. She born and brought up in the proctor medicine at IRM, Salt Lake, under the Professor B. N. Chakraborty. Very nice. She did her master's in clinical embryology from NUH Singapore. She has got the advantage of planning stimulation in IVF as well as being a qualified embryologist, checking the embryo potential of the couple and planning the. Welcome to you, Dr. Konkon Mitra.
Speaking about it in front of this outset, I would like to thank ladies and gentlemen. At the outset, I would like to thank ACE for giving me an opportunity to speak in front of this August gathering. The topic I have been given this evening is to talk on the semen preparation techniques, and I would be speaking about it for the next 15 minutes. Now, coming to a little bit of physiology, we all know that the ejaculate is formed from the concentrated suspension of spermatozoa mixed with and diluted by fluid secretions from the accessory sex organs. Once the ejaculate comes out, the spermatozoa are trapped tightly in a coagulum developed from the proteins of the seminal vesicular origin, and subsequently over a period of time, it is liquefied by the action of the proteins. And a quick recap here, that 90% of the semen volume is formed by the secretions from the accessory organs, and only 10% of it is formed by the sperms. Now, semen is considered as a biohazard, and before it is entered in a lab, we have to screen it for the infection like HIV and the hepatitis. And a quick recap on the sterile materials and the techniques that must be used. Proper cleaning instructions have to be given. In the place we keep the semen containers, preferably a laminar flow. And the previous evening, we should have cleaned the place with 70% alcohol. No lubricants or body lotion should be entertained because they have a spermicidal effect. And all the semen samples have to be properly labeled because it is an embryologist's nightmare when there is a mixing of semen samples, and we all know, I'm sure in this gathering, what is the consequences. We have to collect the semen in a wide mouth container, preferably with a non-toxic material, and that should not be touched in the inside of the container to avoid contamination. Now, the semen analysis is mainly the quantitative, where we see the total number of spermatozoa to assess the testicular function, the patency of the post-testicular duct system, and the secretory activities of the gland, and coming to the qualitative analysis where we look at the motility and the morphology of the sperm. Now, again, a little bit of sperm physiology, where once the semen is deposited in the vagina, it travels through the uterus to the fallopian tubes, and there they get collected at the storage site. And at this storage site, there is a process which calls capacitation. And after capacitation, the sperms leave the storage site and gradually move and get attracted towards the oocyte through a procedure of chemotaxis. And through this chemotaxis, the sperm penetrate the cumulus cell and they bind to the sperm receptors on the egg and undergo the acrosome reaction. Now, what is the purpose of the semen preparation? The purpose of the semen preparation is to remove as much as possible the seminal plasma, because studies have shown that once the sperms remain in the seminal plasma for as little as 30 minutes, the fertilization capacity of the sperms go down. and to achieve as much as possible morphologically normal and motile sperm with very minimum reactive oxygen species which are detrimental to the fertilization and preserving the DNA integrity. So the method of sperm preparation is done to select a highly fecund subpopulation of the sperm. Now, is there an ideal semen preparation technique? Ladies and gentlemen, the ideal semen preparation technique has to be quick, easy, and cost-effective. We have to ensure that there is no damage or any physiological alteration to the separated sperm cell. Now, I will show you a video of a very good semen sample. Here we can all see that the count is good, the motility is good. Now, here is another semen sample where we can see that the motility of the sperms are good, but the count is less. And the third sample is not a very, it's a very poor semen sample where we can see just one or two immotile, mostly immotile sperm and one or two sperms which are just flickering. Now, these three semen samples have to be treated differently. And the most common preparation techniques are migration and the density gradient. The migrations are the swim-up technique, the underlay, and the migration sedimentation technique. And the density gradient are mainly of two types, continuous and the discontinuous, and the filtration techniques. Now, the choice of the sperm preparation methods is mainly dependent on the semen concentration and the motile count on the number of sperms, the ratio between the motile and the immotile count, the volume of the liquefied semen sample, presence of antibodies, agglutination, pus cells or debris, and any kind of infection to the semen. So a few general points, which I'm sure everybody knows here in the gathering, but I would just like to put it in the head that this is extremely important in this stress of era, that for any IUI or an IVF procedure, the timing should be informed well in prior to the patient so that the patient is informed, relaxed when he comes to the lab to give a semen sample. This relaxed patient is an extreme importance because we all have encountered in our clinic where patients come with a very anxious wife lugging behind, asking us a sample we cannot give. So this has to be taken care. There should be a good counseling. The prior viral profile and infection screen should be there in the lab. Nice time of around 30 to 40 minutes should be given for the embryologist and the semen to be kept so that it is nicely liquefied. The pre-wash and the post-wash semen parameter should be notified. The media should be brought at 37 degrees before mixing it with the sperm to avoid a cold shock. And the test tubes have to be labeled ideally with two unique markers, the name of the couple and the unique hospital idea. But in our lab, we use the name of the husband and the wife together. And very, very rarely when the name of the husband and the wife match, we take the name of the place where they belong to. And the record should be maintained. Now coming to a sample which is viscous. The liquefaction in a sample usually takes around 5 to 20 minutes. And this is due to the proteins which are present in the sample. But often we encounter highly viscous semen. And here when we encounter a viscous semen, we usually, when the semen sample is known previously, we give the container to the couple with a little amount of media, which they warm to the room temperature before collecting. Or if we don't, we are not aware of it and we get a semen sample, usually we break and pipette the semen 3 or 4 times or a couple of more times with a 1 ml syringe or with a glass pipette. And I think that does the job. We usually, we never use, in fact, not usually, we never use a chemical in our lab. Now, coming to the liquefaction, spermatozoa gain the ability to move as the semen liquefies, and it usually takes not more than 30 minutes. Now, coming to the details of the techniques, migration, it is the swim up, underlay, and the migration sedimentation technique. The swim up is the age-old technique and the oldest and the mostly commonly used sperm preparation technique. And for this, we need a semen parameter which has a good semen motile sperm, and the count has to be good. And the selection based on the motility, because motile sperms are selected based on their ability to migrate into the medium. In the sperm swim up procedure, low viscosity media is layered over the semen. We allow the motile sperm to swim either up or down into the gradient, separating them from the null-motile fraction, and usually generally yield a population of motile sperm that are used in ART procedure. Here I would like to share a slide with a gathering that we use a normal 14 ml round bottom tube. We have a liquefied semen sample at the bottom of the tube. Very gradually we have the sperm wash medium on the top. And we usually incline it at 45 degrees centigrade, and we keep it at a test tube holder inside the incubator just to increase the surface area of the media so that there is a larger surface area of sperms which will be swimming up. Now, selection is based on motility of the sperm. Sorry, there's a repetition. And here I would like to share a video of the single wash technique. Here, these are the consumables which are used. And this is the sample which looks quite good, not much of a debris. This is a pre-wash semen sample which has been used. And 2.5 ml of sperm preparation media is taken into a conical tube. 2.5 ml. This procedure, this method can be done fast. Then carefully we straight go to the bottom of the conical tube and gradually with a lot of care, we underlay the media with semen sample. Here we have used a plastic tip, but we can also use a glass pipette. And then we keep it inside the incubator in a 45-degree angle for 60 minutes at 37 degree. After that, when we see a very clouded area, which is very rich in motile sperm, we take it out and mix it with a clear, fresh wash media, and we gently mix it up and down. We have to ensure that there are no bubbles formed, not too much of it, because we all know that these bubbles have these harmful ROS. We centrifuge it at 1700 for 10 minutes, and we can see a wonderful pellet and form. We discard the supernatant, leaving the pellet undisturbed. We add very gradually 0.5 ml of the media without disturbing the pellet and still use. And this is a picture of a washed sperm. And we keep it in the incubator at 37 degree till we use it either for IUI or an IVF. Now, this is a swim up method, which is a double wash method. This is again the same, only thing here, we have used the centrifugation 2 times. Now coming to the procedure, a nicely labeled tube. We have kept it for 30 to 45 minutes. It has been liquefied. We checked for liquefaction, the volume, the viscosity. This is a macroscopic examination. Using a plastic tip, we put it at a macler and see. Here the sample is not as good as the previous one. We see a couple of dead cells, but we have ample amount of motiles. We take 1 ml of the liquefied semen in a centrifuge conical tube. Preheated sperm media is used and it is mixed very gently. This mixing has to be very gently, done very, very gently. We mix it up, ensuring no air bubbles are formed, and it is centrifuged with a balance at 1700 RPM for 10 minutes. We see a wonderful pellet again here. We just go down, just take the pellet bit and add fresh media to this pellet, 2 ml of the sperm preparation media, and mix it gently. And this is the second wash we are giving. Here the speed and the duration both are less, 1500 for 5 minutes. And after 5 minutes, we take out and we see the beautiful pellet here. And I'm sure for everybody, this is a delight for any embryologist or an andrologist to get such a semen sample. We discard the supernatant, add 0.5 ml of semen preparation media without disturbing it. And now the semen is put in the incubator and it is ready to use. And we check it when we are using it. And this is quite a very satisfied. We are very satisfied with the semen, which will give us pregnancies. Now, the advantage of this is centrifugation only motile sperm without the WBCs and the dead sperm. So we can reduce the reactive oxygen species. And the disadvantage here is the amount of motile spermatozoa retrieved is relatively low here. Now coming to the second technique, that is the density gradient, which are continuous or discontinuous. Here in the continuous one, we use just 1% of the media, gradient media, and in discontinuous, we use, which I will be showing in the video, is a discontinuous 2 methods. Here, the selection is based on the motility and the side of the sperm, centrifuged through a continuous or discontinuous density gradient of either colloidal or salinized colloidal silica. Here, the motile mature is of a higher density. Here, three things. The basis of this technique is that a mature sperm has a higher density than an immotile spermatozoa, and the leukocytes and the cell debris are also of a lighter density. So they tend to be on the top, and the mature ones are at the bottom. So density gradient centrifugation is the method of choice for low motility sperm samples, poor forward progression sperm samples, large amount of debris, anti-sperm antibodies, and seropositive patients. And high proportion of functional sperms can be recovered from the gradients. So this is a sample where I would like to show. See, this is a sample which is very thick. The count is good, but the movement are very sluggish. A lot of pus cells, a lot of dead cells are there, and rotatory movements. So this is good for density gradient. Now showing the video of the density gradient method. Workstation, the laminar airflow should be on. The temperature should be 24 to 25 degree. We should keep it at the room temperature for 30 to 60. 80% gradient is used, 40% gradient, and a sperm preparation Hippies buffered media is used. See, this is a poor semen sample. The motility is low, the count is low, lot of dead sperms are there. First, we take 1 ml of 80% solution in a sterile centrifuge tube at the bottom. We overlay it with a 40% semen on top. And over that, we carefully put the semen sample, very carefully. You can see the beautiful interface. You can see because of the difference. One is a colorless media and one is a very light pink media. And this is a schematic representation. I'm sorry for the sound, but I don't think I can do anything about it at the moment. And we also keep ready a fresh tube with fresh media. We put the media, we centrifuge it for 1700 RPM for 10 minutes again. Here is the schematic. We get a nice pellet and all the debris, dead cells, spermatozoa, immotile are on the top. And we discard the supernatant. We carefully discard the supernatant, taking the utmost care not to disturb the pellet at the bottom. We aspirate the pellet and mix it with the fresh media gently again, very gently. And then we give it a second wash. This time the speed and the time is again less, 1500 RPM for 5 minutes. After 5 minutes, we discard the supernatant and we keep the pellet without disturbing. and we resuspend the pellet containing the sediment. And this is ready to use. This is what is the density gradient method. And we put it again in the CO2 incubator till we use it. And we are checking it before using. We see that we have enough motile sperm, the dead sperms and the debris and everything are gone with this thing. Now, the advantages of the density gradient, we usually get a very clean fraction of highly motile spermatozoa. Leukocytes and dead cells and WBs are all eliminated, so eliminating the fear of ROS to a great extent. But the disadvantage is that density gradient media is a bit more expensive. So density gradient media is not used, density gradient method, sorry, is not used when the semen sample has extremely low sperm concentration, when it is very highly viscous, and samples which have a very huge proportion of cellular debris. Here, I would like to mention, ladies and gentlemen, we often, when we see this, we put the patient on a course of doxycycline for 3 months for a period of 8 days every month. Now, filtration techniques, I will not go into it because this is something we don't do in our lab. So now coming to a few important tricks and tips, or the basis is that if we centrifuge it for a longer time and a longer speed, there is a recovery of both motile and immotile sperm. So we have to be very careful of the time and the speed. The debris, round cell, abnormal form, amorphous head never reach the bottom because their density is very low. Gradients with a larger volume improve filtration, but they decrease yield because the sperms cannot move and travel. Severely asthenospermic samples with a normal sperm density but poor motility can also be distributed in small mini gradient aliquots. So we mix at the end total, and we often pre-mix the gas mixture with CO2, O2 and nitrogen for 90 seconds to maintain the pH, which is again very important. So these are studies which have shown that the temperature of the sperm preparation place and the media, the pH of the place, the preparation techniques, the incubation conditions are very important to maintain the DNA integrity of the ejaculated human spermatozoa. So ladies and gentlemen, we have to be very careful in the andrology lab to take all the precautions with the lighting, the pH, trying to reduce the ROS and keeping the temperature as congenial as possible. And here is a last line which shows the magnitude of problem, and workers have shown that approximately every 4th couple is experiencing infertility due to DNA fragmentation now. So it is quite scary. So here I would like to thank all of you for a patient hearing.
Thank you so much, madam, for the detailed semen preparation techniques. Can we have the next speaker, please? We'll take the questions after that. So our next speaker is a very well-known personality, Dr. S.S. Vasan. He need not any introduction. He's a very well-known man. He has done his MBBS and DNB surgery and DNB urology. He has done his fellowship in andrology and incontinence from Singapore. He is the head of andrology fellowship program and teaches Rajiv Gandhi University for Health Sciences Andrology. He is the co-founder for Janani, a startup transforming fertility management. He is the president of South Asian Society for Sexual Medicine, member Stem Cell Task Force, IISC, Indian Institute of Science. Sir has lots of patents to his credit, and right now Sir is working on the design, development and evaluation of a handheld shock wave generator for biomedical application. Welcome to you, sir. It is a pleasure to have you here on the board. Thank you.
Good evening, everyone. I'm Dr. Vasan. I'm going to give you a brief insight into devices which have a clinical significance in andrology. So let us discuss the various aspects of this. And as we are all aware, at this point in time, male infertility diagnostics consists of the basic semen analysis, both macroscopic and microscopic, and we have certain specialized advanced tests which has been evolving over a period of years. And some of these are the sperm DNA fragmentation, SCASA, tunnel, comet assay, ROS, and you have the oxidative reduction potential, which is the meioxis, which is the new baby of the block. And we had some zona pellucida functioning tests, hemizona binding assay, acrosome, and so on and so forth. So consequent to this, what are the various advances which have happened over a couple of years? We will discuss now, and that is the aim of this lectures. Some of the advances which are happening now are sperm analysis emerging techniques or semen analysis emerging techniques using digital holography, super resolution microscopy, NGS. The traditional technologies used for sperm sorting, such as density gradient centrifugation, swim up, comet assay, we all know that cannot select sperm with optimal DNA quality. So what has changed in the last couple of years is there are technical advances which have improved the accuracy, speed, and efficiency in the analysis of sperm motility, morphology, and genetics. Also improved sperm selection for infertility treatment by measuring the sperm behavior and biomarkers of fertility potential. There have been discoveries in sperm locomotion and behavior to improve the understanding of sperm motion and significance in the female reproductive tract, and analysis of sperm structure and molecules such as Caspar channels and ubiquitin, which have evolved, which helps us to understand sperm physiology and contributes to identification of biomarkers of potential. So the basic progress in sperm analysis promises non-invasive testing for sperm selection, increased efficiency and accuracy via automation and artificial intelligence, and improved access of point-of-care assays. This is what is the major change. What are the key discoveries in this area? We have discovered that surprisingly sperm were found to move along in helices and chiral ribbons and swim in a slithering mode within a confined space. They also exhibit behavior such as boundary following, corner turning in microchannels, which improves our understanding of sperm locomotion within the female reproductive tract. To overcome the diffraction limit of light microscopy, super resolution microscopy has been used to reveal sperm ultrastructures such as proteins which are involved in motion regulation and acrosome reaction. Similarly, using immunostaining and mass spectroscopy, biomarkers have been identified such as ubiquitin, TEX101, HSP-A2 that indicate sperm abnormality, obstructive azoospermia, and sperm maturation respectively. So let me go through some of the advances which have happened. We know that sperm quality testing involved basically testing for DNA fragmentation and ROS using various devices. And now we know that there have been several techniques. We know that some of the gold standard techniques are your tunnel assay and your comet assay or your SCASA, which uses a flow cytometer. Similarly, ROS measurement have undergone a change wherein we were looking at measuring oxidative stress. Now we measure the redox potential, and the oxidative stress measurement used to happen with a luminometer. Now redox potential measurement uses an instrument called meioxis. So sperm quality testing has further improved because we have recognized out many cases of fertilization failure, which happens or implantation failure occur as a result of genetic imbalances prompting development of fluorescence in situ hybridization or FISH technique to detect sperm aneuploidy and abnormal number of chromosomes. We know that by tagging FISH, we have been able to identify numerical abnormalities involving chromosomes 13, 18, 21, X and Y. And this has been a significant development. The next evolution has been in molecular and epigenetic markers using NGF. We know that more than genes, more than being a simple carrier of male genetic material, spermatozoa supply epigenetically modified genome with RNA and protein components critical for fertilization and embryonic development. The sperm epigenome is characterized by DNA methylation, which modifies genetic material, and extensive protemination or DNA repackaging. We also know that now proteins 1 and 2, P1 and P2, replace histones during spermatogenesis, leading to a more compact chromatin packaging structure necessary for sperm function. The relative concentrations of P1 and P2 normally, which is equally expressed, may be abnormal in some groups. So the basic research has been in molecular and epigenetic markers, basically aberrant DNA methylation, wherein we detect hypermethylation, which is, and the gene which is responsible is the cyclic AMP response element modulator, or the Sperm gene, which is one of the classical examples of hypermethylation, as well as the MEST, which is the mesosome specific transcript, and your P1 and P2 concentration, which are markers. Molecular and epigenetic markers have also been studied by NGS, which is able to identify coding and not coding RNAs, including thousands of microRNAs. We know that now microRNAs are involved in regulation of early embryogenesis, and there is a brilliant paper by Salas and Hutos, which demonstrated difference between microRNA profiles between fertile and infertile men, finding defect-specific altered microRNA profiles in groups of infertile men with isolated oligospermia, asthenospermia, or teratozoospermia. The evolution in proteomics has happened both for oligoasthenospermia as well as for azoospermia. We know that the semen concentration only constitutes 10% of the total ejaculate volume. 90% is seminal plasma, which contains a diverse molecular composition. And there have been several molecules which have been identified: olfactory receptor 5R1, lactoferrin, HCAP18, spindlin1, and clusterin, which may be required for male fertility. Similarly, in azoospermia, there have been differentiating azoospermia from obstructive to non-obstructive based on seminal plasma protein biomarkers has been advancing at a rapid pace. And also we have both at the testicular level and the seminal plasma level. Seminal plasma is advantageous as it's readily available, whereas testicular samples requires a biopsy. So you have these various biomarkers: TEX101, LDHC, ACRV1, ECM, PGD, DS, etc., which are markers in the case of non-obstructive as well as obstructive azoospermia. And depending on the threshold and sensitivity, you are able to identify obstructive versus non-obstructive azoospermia. The other research which has been going on is not only to perfect functional studies and also to predict ART success. Presently, elevated sperm DNA fragmentation is the only test which may be suggestive of a higher risk of pregnancy loss with an IVF and ICSI. Petit and colleagues noted elevated serum aneuploidy rates among couples with repeated ICSI failures, and we already discussed protamine 1 and 2 are necessary for compacting chromatin, and these two proteins are typically expressed in similar concentration. So men undergoing standard IVF, those with abnormal protamine 1 and 2 ratios, were found to have reduced fertilization rate compared to men with normal ratio. So to summarize, sperm analysis enabled applications have basically focused on sperm motility measurement, which has been traditionally measured by manual estimation. Then we had the computer aided sperm tracking, which is more accurate and consistent. Similarly, morphology, which has traditionally used by a fixation and staining method, which damages the sperm, destroying the head and also the sperm in terms of its potential to fertilize. So now we have non-invasive morphology measurement, which uses the DIC imaging to increase sperm contrast, image reconstruction, and processing of automated measurement. So, and 3D morphology measurement. So interferometric phase microscopy, by reconstructing various things by a phase map to reflect actual optical thickness of the sperm. So we are using more and more techniques are coming in where we are using non-invasive morphology measurement, 3D morphology measurement. Similarly, some of the advances are in your sperm DNA fragmentation. We have the SCASA, tunnel, SCD, cometastasis, which are commonly used, but again destroy sperm function. So we're looking at non-invasive DNA fragmentation measurement, which is the Raman spectroscopy, which extracts DNA information from inelastic light scattering by molecular vibration. Chromosomal aberration measurement, which is your FISH, which uses DNA probes to identify chromosome abnormalities, but only can measure a few chromosomes. Genome-wide chromosome aberration, which uses ACGH and NGS to provide genome-wide sequencing. Telomere measurement, which uses quantitative FISH DNA methylation analysis, which is a microarray, and NGS for methylation pattern analysis, histone modification analysis, which is a chromatin immunoprecipitation using protein-specific antibodies, and your RNA analysis, which uses real-time PCR RNA sequencing for various analysis. The next area of evolution has been, one is to diagnose, one is to find efficient sperm. And we have traditionally looked at selecting sperm by motility and density, where we have used swim up and gradient centrifugation, and also by morphology, wherein we have used IMSI, wherein we have looked at higher magnification to select the sperm with normal subcellular structures. But we know that this is time consuming. So consequently, there have been various techniques which use non-invasive measurements or machine-based, which are sperm sorting by racing motile sperm inside microchannels, which is microfluidic devices enabling collection of motile sperm at the outlet, using boundary following behavior to select sperm with low DNA fragmentation, which is your microfluidic device, which branch microchannels, sperm selection by using fluidic flow to induce rheotaxis, which is your microfluidic device with fluidic flow and geometric constraint, using chemical gradient to select sperm with chemotactic behavior, and using hyaluronic acid as a physiologic selector of sperm, and your annexin V used to separate apoptotic sperm. So these are all techniques which are evolving, which helps us to get quality and competent sperm instead of using. So these are some of the summary of the various information I have provided, which is your DNA fragmentation, which is your analysis of your sperm genetics, wherein we are using Raman spectroscopy acquired, which is the cell membrane, acrosome, which is the blue line, nucleus, which is the red line, and midpiece, which is the yellow line. So the spectra uses these imaging to reconstruct and predict the damage to the sperm. Analysis of sperm genetics uses genomic hybridization, and here this is a comparative genomic hybridization, wherein the sperm DNA from a patient and a normal control are labeled using fluorescent dyes and mixed together and hybridized on a microarray platform. So analysis of sperm genetics also is possible by using the genomic DNA, fragmenting and sequence capturing, and then having a DNA sequence. So in this, we fragment the DNA to small stretches of 50 to 500 nucleotides, and billions of DNA fragments are then captured in an array and analyzed in a single reaction. Analysis of enabled microsperm selection using microfluidic. Here it's a microfluidic device to start sperm by exhaustion of the sperm inside the microchannels. And you have analysis which is enabled sperm selection with a lower DFI, or the DNA fragmentation index, by using a microfluidic channel wherein the sperm uses its curvilinear or rotational movement, and you capture the sperm. Similarly, apoptotic sperm selection is enabled by using a magnetic activated cell sorting, which is what is the technology which is available, which is called MAX. So there have been several areas of interest, as we all have seen over the last couple of minutes which I presented. One is looking at how do we perfect the technology of analysis of the sperm in terms of motility, morphology, how do we perfect it, and then looking at specialized testing, how do we do the specialized testing, which gives valuable clinical information, both at the level of the DNA, as well as in terms of proteomics, as well as in terms of fertilizing ability, and then looking at other factors which may enable as biomarkers in our research. So where are we today in this journey? In terms of sperm analysis, these are the various discoveries which have happened. Now we can have a flagellar tracking technique, wherein flagellar tracking beat consisting of two bending waves and sperm navigating through this phase between two harmonics. And so this is a perfection in the technology. Both from the flagellar tracking can also be used in rheotaxis, and people have used 3D to analyze the slithering or the swimming mode of the sperm. And then you have opsin proteins located on the sperm membrane function, which acts as a thermosensor in sperm thermotaxis. 5% of the sperm swim in helicals, and majority of the sperm prefer right-handed helices. And this is what the digital holography has shown. Sperm swimming speed is positively correlated with spinning speed in 3D, and 100% of the sperm show right-handed spin with head axis. So all these developments will aid in further clinically understanding how does the sperm move in the reproductive tract. So consequently, these are all exciting. So similarly, the analysis or the discoveries in terms of structure, we know that mitochondria are helicoidally distributed around the axoneme with midpiece. Caspar channels are normally distributed in linear quadrilateral domain along the flagellum. And then in midpieces, actin forms a double helix that accompanies mitochondria with periodicity of 214 nanometers. CD46, a membrane cofactor protein, undergoes dynamic reorganization during the acrosome reaction. SUN4, a spermatid nuclear membrane protein, is essential for nuclear remodeling, and so on and so forth. So as you can see, this is a long list where exciting discoveries have happened using techniques like SNORM, STORM, STED, SIM, immunostaining, mass spectroscopy, and consequently we are in an era where we are able to diagnose some of these problems, and all these research will aid in for the clinician to enable or understand or bring out technologies which will aim in perfecting, one, whether we can analyze the sperm in terms of its fertilizing potential. Two, can we analyze the sperm in terms of its defective potential? Third, can we predict the sperm in terms of its fertilizing potential? Four, in terms of obstructive and non-obstructive azoospermia, can we have techniques which non-invasively detect the sperm? Again, in all this, the whole emphasis has been on trying to develop techniques as least invasive as possible. As you can see, even in an IUI, can we eliminate the margin of error which is associated with the technician? So this is an exciting era, and this is an era where we will see a lot of new discoveries which will come in the next decade or so, and consequently, we'll have to look out for various things which will develop in this field. Thank you very much, and thank you for your patient listening.
Thank you, Dr. Watson, for the wonderful talk, and thank you for enlightening us with the recent advancements. So now we have the live session by Dr. Watson on basic semen analysis and other current devices used for semen analysis, sperm function tests like HOS test, assessment of DNA fragmentation index, morphology staining, and etc. So over to you, tech team. Please take us to the live session.
Hey everyone, I'm Dr. Vasan, a uro-andrologist, and I'll be now playing a video which demonstrates the various aspects of semen analysis, including Horse test, morphological staining, DNA fragmentation assessment, reactive oxygen species assessment, and some amount of computerized semen analysis. This video was prepared by my team consisting of Pooja, Rabia, and Pratibha, and I would like to sincerely acknowledge them for their wonderful effort. Now let's see this video and understand the various nuances of a proper semen analysis and advanced sperm function testing. Thank you.
Once the sample is received by the patient, keep 30 minutes in the incubator for the liquefaction. Then take the sample, check with the patient name, age, MRI number, take the sample for processing. Let us check microscopic examination. First comes the value. Check for the patient sample value. Then check for the liquefaction. Check for the pH. Once the sample is analyzed for microscopic examination, write the value, liquefaction and the pH. So microscopic examination is done. Let us do the microscopic examination. It means checking for the sperm count, motility, take 10 microliters of liquefied sample, place it on the macro chamber, fix the cover slip, observe under microscope. First, we'll count for concentration, so now the concentration is done. Note down the sperm concentration. Take a clear glass slide, place it on the surface, take 10 microliters of sample, place it. Prepare a thin smear for morphology. Label the smear with patient name, then air dry the smear. Once the smeared slide is dried, we'll start the staining procedure. First, fix the slide in the fixative for 10 minutes. After 10 minutes, take out the slide. Just remove the extra fixative. Fix it in second stain, that is eosine. Fix the slide in eosine for 5 minutes. After 5 minutes, remove the slide. Remove the excess stain. Fix it in the counter stain, that is methylene blue. Fix for 5 minutes. After 5 minutes, remove the slide from methylene blue. Remove the extra stain. Wash the slide in the running tap water or with the distilled water, drop by drop. Just clear the background. Place the slide for air dry. Check for the morphology assessment in oil immersion. Check for the normal forms, head defects, mid-piece and neck defects, tail defect, acrosome. And for ERC.
HOS test, that is hyperosmotic swelling test. HOS test is done to detect the integrity of the cytoplasm of the sperm. So this procedure involves (40 microliters of HOS solution, and then 10 microliters of semen sample is taken together and mixed well. Once it is mixed properly, keep it for incubation for 10 minutes. After 10 minutes, place 10 microliters of the sample cover it with the cover slip, and then we are focusing at 40x. So when we focus on the microscope at 40x, we observe few sperms which have coiled tail and few sperms which have bulged head. It just means that the coiled tail, the sperms which tails are coiled, they are live, and the sperms which heads are bulged, they are detected as dead. So the percentage of coiled sperms are detected as live, and that is the HOS value which we keep. We counted for about 100.
And I'm going to explain about the DFI staining. First, for DFI staining, you have to make a smear. Take a glass slide. You need to mention the patient's name over here, like it should be visible, and it should be written by marker so that it should not get washed away. So first, I'll take a 10 microliter of semen sample. I'm going to make a smear and keep it for 10 minutes for air dry. After air dry, first we will take fixative, place it inside the container for 10 minutes. After 10 minutes, take out the slide from the fixative then transfer into aniline blue for 10 seconds. After 10 seconds, take out the slide and wash away the stain from the slide. After taking out the stain from the slide, keep it for 30 minutes for air dry and then view under oil immersion 100x. The evaluation of alanine blue stain indicates that if the sperm head stain dark blue color, that means it is an immature sperm, and if the sperm head takes light blue color, that indicates it is a mature sperm. Thank you.
Hello everyone, good afternoon. This is the sperm chromatin structural assay, which we are taking as a sperm DNA fragmentation index. Firstly, this is our flow cytometry, which is a standardized technology for DNA of sperm. So mainly this equipment is based on the blue laser light, which is of 488 nanometer. Acquisition of the sample is necessarily a flow cytometry equipped with a blue laser and minimum of 2 fluorescence parameters such as green, which goes for 530 nanometer, and red, it goes for 650 nanometer on the blue laser light, which will be present inside the machine. AO signal, it is a acridine orange signal from double stranded DNA was collected using 527 to 30 nanometer filter, and that from a single stranded DNA was collected using 675 nanometer filter. At least 5000 counts events are collected per sample at a low flow rate of 0.1 to 0.3 microliter per second, and single samples will be analyzed for thrice, and then it will be analyzed for mean and SD. The sample is, once the sample is collected, sample will be incubated at 37 degrees centigrade for 30 minutes, and sample will be checked for the concentration and the given concentration, and the concentration is calculated with certain measurement, and it will be diluted with TNE buffer, and then goes with the dilution for acid denaturation of DNA at site of stranded breaks, and then AO staining of single stranded, red color, and double stranded with green color DNA. Now the sample is diluted with first with DNA. The sample is diluted and it is ready to run the sample. In the machine we will be having a probe which will suck the sample, and the machine should be first cleaned with a decontamination solution to remove the excess of stains which surrounds the pipes and the interwall of the filters. Then it will be cleaned by a cleaning solution just to clean the area once again. Then the prime acquisition will be given to remove the excess of stains again. Then the sample can be run, and the sample will be in the speed of 0.7 to 1 microliter per second. It will be run by the certain gates, locks, and as well as the certain parameters which is as FSC size, FL1, FL2, FL3. So the cleaning, after the cleaning procedure is done, the machine will be run with sheath again. just for 5 to 10 seconds. Just to rinse the area once again. Then the samples can be run. This is the sample which is diluted with TNE, acid detergent buffer, and with our acridine orange stain. Push the sample tube very gently. After inserting the sample, the speed can be changed to 0.1 to 1. As mentioned before, it can be run for 50,000 events. Sample is run. Save the sample. Single sample will be run for thrice to analyze and take the mean and ST of the sample. And for every single run there should be a self-clean. Mix the sample and insert it. Mix the sample for third time. So now the sample is run for thrice and it can be analyzed for mean and ST. Thank you. It's STA Vision. It's a sperm quality automated analyzer which gives the sperm concentration, motility, morphology, everything automatically. The system will be cleaned as per the daily maintenance. Then the test, after the sample is collected, the sample will be kept in the 37 degree for 30 minutes, and after the incubation, the sample will be checked for the liquefaction, pH, then the liquefaction is done. Just check for the pH. The test patient details has to be mentioned in the system. So let us give the patient ID, patient name, sample ID, patient age, and the abstinence should be mentioned. The volume of the sample is measured already. The sample volume has to be mentioned, and the percents, pH, appearance of the sample as it normal, moderate or viscous, and the viscosity should be mentioned, and the liquefaction also should be mentioned. After entering all the details of the patient, give test now and we need to wait. The system is calibrating, and we are not supposed to insert the capillary when the calibration is taking place. Meanwhile, let us collect the sample in the capillary without any air bubbles. Now the sample is collected in the capillary. Be aware that no bubble should be formed inside the capillary. And we need only 0.5 ml of sample to insert. Now the machine is asking for the insert capillary into the chamber. Let us insert the capillary. So test is in progress. Do not move the capillary or the device during the test time. Meanwhile, let us have a clear slide. Mix the sample gently. Pipette 10 microliters of sample. Place in the tip of the slide. Cover the cover slip. So now the test is done. The machine is importing the test to the system. The test is transferred. Now, to visualize, insert the slide to the slide rack and place it on the analyzer. Focus the slide. Now need to give the debris has few, moderate, many, or it will be for gross. And there will be a few examples of everything. Just click the examples, select the debris field. So we are finding or refuse or debris which can be given as few or. Hello everyone, my name is Pratibha. I am explaining about the reaction oxygen species measurement using the luminometry analyzer. So luminometry analyzer, it is only for the oxygen reaction species will be measured under it, and we can also measure the extracellular and intracellular production of ROS. Once a sample is collected, semen sample is collected, sample will be incubated at 37 degrees Celsius about 30 minutes. Then the sample will be checked for the microscopic, the volume, liquefaction, everything will be checked. Then the test will be performed immediately. So RS determination is should be set up in the luminometer. I will show you the luminometer. Let us see the automated analyzer, luminometer analyzer. The test should be performed in the dark. The luminol will be placed inside the machine. It should be in the dark surface. Take 40 microliters of semen sample. Add 5 microliter of luminol, which will be in the dark surface. Add luminol to the 40 microliter of sample. Do not mix. Directly measure the test. So just give the measure luminescence. Analyzer automatically gives the result. After After the reading comes, just remove the bullet immediately. Then the result can be given to the patient. Thank you.
So this is the home collection sample. While collecting at home, the patient will be given with a transfer media, that is zapnil media. After collecting the semen sample, the patient is going to add the zapnil media, which consists of 2 ml in a vial, is added into this container. Then it is transferred to the lab. Once after the sample is received at the laboratory, the sample is incubated for 10 minutes, and then after 10 minutes we are going for analyze. So after incubation, the very foremost thing we are going to do is we are going to check for viscosity. Then volume. Then we'll check for the pH. So these are all the microscopic evaluation. Then we'll go forward for the microscopic evaluation. So microscopic evaluation includes concentration, motility and morphology. So 10 microliters of the sample is loaded on the macular chamber. Initially, while preparing the morphology slide, let's label the slide. Drop a drop of semen sample on a clean glass slide. Then give it a thick smear using another clean glass slide. So then allow it for air dry. Meanwhile, while the slides gets ready, so this is the S-moti media. This is the recovery, we also call as recovery media. This S-moti media is added to the semen sample which we have received from the patient. An equal volume of S-moti media and semen sample which contains transfer media is added. Let's mix both of them gently. Then allow it for incubation and then record the results accordingly.
So thank you, Dr. Vasan and team of Dr. Vasan for demonstrating the procedures very beautifully. So now I request tech team that if we have any questions, we can take the questions or we can proceed to the next session. If you're not having any questions, you can present me the CV for the next presenter and proceed with the next session. So now I would like to welcome Dr. Sunil Jindal. He's a consultant andrologist and scientific director, Jindal Hospital and Fertility Center. He's a gold medalist and best postgraduate in MAMC New Delhi. He has received around 35 awards from different organizations and have collaborated with multiple institutes for research, including American Center for Reproductive Medicine and National International Offline Online Master Trainer in Surgical Sperm Retrieval. He has various publications and various book chapters, especially even a part of special interest group in fertility enhancing endolaparoscopic and microscopic andrology surgeries and special interest in surgical, medical, and laboratory andrology. Welcome, Dr. Sunil Jindal, to this workshop. So over to you, sir. So you are on mute. So you are on mute. Yeah.
Thank you very much. Thank you very much, the ACE team, which are doing an amazing job. Thank you, Dr. Ratna, Dr. Gautam Kastagir, and the whole team for inviting me. And of course, thank you very much. The people who are listening, you could be doing anything else during this time, but you decided to be over here in this program. And I hope I'm able to do something which is worth your time. So keeping all this in mind, there are 5 things which I'm going to discuss and cover over here. The first thing is the different types of azoospermia and what are the modalities for that. What are the better modalities for obstructive azoospermia? What are the modalities for non-obstructive azoospermia? How can you improve the sperm harvesting in non-obstructive azoospermia? And number 5, how can your laboratory beautifully partner with you to have a good result? So for these 5 things, if you give me the permission, I'd like to share a small story of my life with you. I was in Maulana Azad Medical College. I did my MS from there, my registrar job from there. And there was no separate urology department. There was a urology department which was attached to the general surgery department. I incidentally was the chief's PG and then the registrar. So the urology department was always there, and there was a department of andrology headed by a very famous andrologist, Dr. Ramchandramurthy Kaza. Now, Dr. Kaza would get hold of andrology patients, open their testes, sit with the microscope for 3 hours, and the assistant would have a tough time looking into the microscope and pouring water. And after 2.5 hours, he would find a sperm and know nothing what to do with it. So this used to happen with us a lot of times. It even happened with me. And after 2.5 hours, when he said, I found a sperm, and I told him, sir, what will you do with it? Because there was nothing to do. But only when we started ICSI in 2004 did we realize that the dots are getting together, they're joining, and these sperms can be beautifully used. So all the experiences which I have about andrology, surgery, laboratory medicine, I'll just try to put them together in 30 minutes and present them to you. So I'll just begin my slide. And can the slide share be seen?
No, sir, not yet. Yeah, now it's fine. Now it's fine, sir. Now it's
okay, fine, great. Could the team spotlight me? Because I just have a single window to see myself. Could you spotlight me, please? Thank you. Now, surgical sperm retrieval, we need to understand 2 things about azoospermia. 1 to 3% male population affected, 10 to 15% of male infertility. Massive number. Nobody tells each other they have this problem. Otherwise, it would be known to more people. 2 types of azoospermia, obstructive and non-obstructive. Obstructive would have a normal sperm production, mechanical blockage, vasectomy, post-infectious. And non-obstructive, we definitely know, is because of sperm production deficiency, chemotherapy, cryptorchidism, and so many different reasons. But the first thing which we need to do before a retrieval is to know exactly what the problem is. And to know that, you need to at least mentally categorize what does the patient have, an obstructive or a non-obstructive azoospermia. So an obstructive azoospermia will have a normal testes, a normal endocrine profile, and a mechanical blockage. So the spermatogenesis is going to be normal. But in non-obstructive azoospermia, if we go into spermatogenic failure, the FSH LH is going to be high, testosterone is going to be low or normal, and the testes will be small or normal, and this spermatogenesis is disrupted. And these disrupted patients are the patients who actually bring in a challenge. So if the volume is more than 1 ml and the pH is more than 7, if the FSH is high or low, you could consider them to be either hypogonadotrophic hypogonadism, or if there's an anabolic steroid, the FSH will be high, low LH and high testosterone. But if the FSH is high, LH is normal or low, testosterone is normal and low, it becomes a non-obstructive azoospermia. And of course, for obstructive azoospermia, the testes is of a good size and the FSH LH are going to be most probably normal. So once you have distinguished this, then you need to decide what do you want to use. So in case it's an obstructive azoospermia, normally there are two methods which are used. One most commonly used is PISA, percutaneous epididymal sperm aspiration. And the other one which we normally use or could use and sometimes use is MISA, microsurgical epididymal sperm aspiration. So why would you use a MISA over a PISA? I'll let you know in the next slide. And in case you have not been able to retrieve a sperm in an obstructive azoospermia by PISA, you would like to do a TISA. A TISA would be testicular sperm aspiration. But normally in a non-obstructive azoospermia, you would need to do a TISA, which is a testicular sperm extraction, or a micro TISA. So the advantages of a micro TISA are phenomenal, but there are a few other techniques close to a micro TISA, which I'm going to demonstrate to you and also show you in a video. So that is going to make the whole picture complete. So you use, this is a PISA, and in a PISA, what you normally use is, I'll just, first of all, before this slide, I'll just talk about a PISA. A PISA, you would use a 24 or a 21 gauge needle. You could use a 1 ml or a 5 ml syringe. In a TISA, you'll use a 17 to 19 gauge needle with a 20 ml syringe. Rita normally we do not do, and a testicular biopsy I'll show you, and a micro TISA I'll show you. So what do you do in a PISA? So there are two methods of doing a PISA. The first important thing is you've got to hold with your two fingers the epididymis, get hold of the most turgid area and know exactly where your needle is going. You need to have a little bit of media in the syringe, and you need to give a gentle suction. Let me be honest with you. In an obstructive azoospermia, the moment you hold with your fingers, you know this is the area which is turgid, and the moment the media gets stained slightly whitish, you know that you've got sperms, or at least some kind of a fluid. So now you're going to send it to the laboratory. This is how an obstructive azoospermia epididymis will look like. You can see turgid, and you can see absolutely full epididymal tubules. Now, testicular sperm aspiration or PISA, which at times when it is not successful with a BD syringe, you would like to use a 21 gauge needle, a butterfly. Now the tubing is going to be filled with media, and you're going to use a 10 ml syringe to put a pressure. And of course, the moment something has come, you're going to clamp it. Open the clamp, and whatever you have aspirated, you're going to send to the laboratory. So this is the second technique which you may use if you're not able to get with a BD syringe. Now, how would you process? I have brought in these slides because this is very important. So you would homogenize with sperm medium to prevent any agglutination. We all know that. You'll inform the surgeon immediately, and I'm talking from the point of view of an embryologist now. You'll inform the surgeon immediately that sperms are seen. The surgeon is going to identify the site, the side, and the point of retrieval on paper. Now you can use the native sample, diluted in sperm medium. you need not wash if the sperms are adequate. And you could load about 1 microliter of the sperm suspension in PVP if the motile sperms, you have motile sperms with progressive motility. This is where you would load it directly into the PVP. You can load it if it's good. We may need to incubate it for about 10 to 20 minutes so that the motility may come into these sperms. Now, in case there is a low motility and the sample is contaminated with cellular debris, you could load about 4 to 10 microliter of sperm suspension on a spread which you have made with sperm medium on the plate, which you can very well see over here. So once you have put and inoculated this with the sperm suspension, any motile sperms which flow towards the periphery can then be picked up, immobilized, and transferred to the PVP so that you could immobilize, aspirate, and inject into metaphase 2 oocytes. So another important thing, please preserve these sperms and please do a cryopreservation if you have anything left over. So now I'll talk about obstructive azoospermia and TISA. You would use, it's not a 21 gauge needle, it's a misprint. You would normally use a 16 gauge to a 17, 18 gauge needle. And you're going to use a 20 ml syringe. And what you normally would do is that once you have this media in the syringe, in the tubing, you're going to place the needle in a full area, I mean the anterior medial area, where you do not suspect any blood vessels. And you would like to get as much of testicular seminiferous tubules as possible. You would take these out and then they would be processed further. Now, a TISA could be done under local, could be done under GA. A needle is inserted anteromedially, I've told you. And due to negative pressure, sometimes a seminiferous tubule may come out. And if it comes out, it's exteriorized and pulled out with a small forceps. Now, the processing, once again, mechanical dispersion, what we use, examine the homogenate, inform the surgeon, aspirate and transfer the cell suspension depending on the sperm which we get, dilute the sperm media, wash, aliquot, consider freezing. Once again over here, as you can see, we have a spread. Now, sperm retrieval, these are a few studies I want to, beautiful papers. Sandro has this amazing paper in which the success in congenital absence of VAS is about 100%. And this post-vasectomy is about 95%, post-infectious 100%, about 97 to 98% sperm retrieval is present. Now, what is the outcome? They have compared the ejaculated sperm to the epididymal or the testicular sperm. And just look at it. The fertilization, along with the pregnancy rates, the total sperms which have been retrieved, and the miscarriage rate are commensurate with an ejaculated sperm. So in obstructive azoospermia, the results are very good because the woman normally does not have any comorbid conditions and she's just waiting for these sperms. So these eggs which are waiting for these sperms in obstructive azoospermia give very good results. Now, if you compare obstructive azoospermia with fertile donors, there again you would find that the results are about the same as a fertile donor. So now let's come into the important part of non-obstructive azoospermia. So you would like to do, of course, a cryopreservation. You would like to spin the semen and you would like to see if there's true azoospermia or cryptozoospermia, and you'd like to take at least 2 samples. Now, I would not like to go through the algorithm of this because it's going to take a lot of time, but I'll only say over here that once you have known there is non-obstructive azoospermia, you would like to get a few genetic tests done, then decide, depending on if the patient has had a biopsy earlier, of what kind of results would you suspect. So in practice, normally you would get about 3% hypo, 36% obstructive, and 61% spermatogenic failure. Now, you need to counsel them. Now, these patients always ask you, what are my chances? So the chances per se, if there's cryptorchidism, is about 50 to 70% if the testes have been brought down. I mean, I'm not talking of abdominal testes. Post-infectious, about 60%. Torsion, more than 50%. Post-chemotherapy, 25 to 75%. Genetic, about 0 to 60%. And idiopathic, about 50%. The FSH level, testosterone levels, and testicular volume, they give us a fair idea between the obstructive and non-obstructive azoospermia. However, the importance is that any level of FSH normally would give us an idea of how difficult the sperm retrieval would be, but does not say that sperms cannot be retrieved. Now, this is a very interesting slide because in case histology is there in non-obstructive azoospermia, in cases of hypospermatogenesis, you could retrieve sperms in about 100%, in maturation arrest about 40%, and Sertoli only about 20%. But this should be counseled to the patient before we go ahead. Now, genetic causes, Y chromosome microdeletion, if it is done, and if it is in the AZA or B area, no sperms would be retrievable. However, if it is in the AZC area, sperms would be retrievable, but the children, in case it's a boy, will have the same problem. So you need to counsel. So I'm going to tell you a very interesting story of the patient of ours. He came to us, Y chromosome microdeletion, did a micro-TSA. He had 2 babies. He had 2 girls. So he left very happy. He came back after about 3 years and he said, I want to try again. So I told him, in case you're very lucky you've had girls. If you had a boy, you would probably have a boy who would have the same problem as you. He said, please try again. Whatever I will have, I will have. And we did an ICSI again. And he had 1 boy and a girl. And you know what he said? He said, if today, agar aaj, mere bachcha ho sakta hai, तो उस टाइम पर जब मेरा बच्चा बच्चे के होने के लायक होगा, उस टाइम पर आप क्या सोचते हैं? कुछ नई चीज नहीं आई होगी? And it's interesting, but in spite counseling, the need for a baby, to have your own baby, lets you have your own baby. Now I'll talk about the management of non-obstructive azoospermia, the selection of the sperm retrieval candidates who may benefit, and what you need to do. So the most important thing is that the intratesticular testosterone should be at least 300 nanograms per deciliter. Because if it is less than that, spermatogenesis will not take place. Now, to optimize the testosterone in these men becomes very important. I normally do it. There are a lot of studies which show that hormonal therapy might increase, but they're all low quality studies as per se. But the important thing for us to understand is that in a person who has low testosterone, you first need to bring the testosterone to normal till you do a micro-TSA. So he was another patient who we needed to treat before we got sperms. But the most important thing for all of us is to understand that whenever intratesticular testosterone is brought to normal by HCG therapy, the chances of spermatogonial DNA synthesis is increased. So what do you normally do? You normally would like to see 2 things. You would like to see the testosterone. You would like to see the TE ratio. In case the TE ratio is less than 10, then you would like to give these patients aromatase inhibitors. In case the TE ratio is normal and the testosterone is less than 300 nanograms, you would like to give these patients HCG. And once you've given HCG, sometimes what happens, the FSH goes down. And if the FSH goes down, then you have to add HMG or FSH so as to bring both the levels to normal before you do a retrieval. Well, there are a lot of studies now. So in case there is a varicocele in non-obstructive azoospermia and you can wait, you could do a varicocele repair before you would like to go ahead and do a sperm retrieval. Now, I'm just bringing out the literature. When you do a TSA in non-obstructive azoospermia, the retrieval is in terms of 15 to 50%. In conventional TSA, it's about 20 to 60%. And microdissection testicular sperm extraction, about 40 to 67%. So micro TSA has actually 1.5 times more likely to result in a successful sperm retrieval than a conventional TSA. And if you look at this particular paper, which was a very large paper which was published, that the overall percentage is from 45 to 25. In hypospermatogenesis by micro TSA, you can get sperms in about 93% compared to 64%. In maturation arrest, about 64% compared to 9%. And in Sertoli, only 20% compared to 6%. So this is a very big number. Well, in case the histopathology does guide us at times of the percentage of sperms we're going to get in them. And in hypospermatogenesis, 91%, maturation arrest 52%, and of course, Sertoli only syndrome about 21%. But sperm retrieval on the whole would be about 55.8% in case we're using a micro TSA. This is a very interesting patient, and this is when our journey started. And this journey started when he came to us, and he was a childhood cancer survivor, Hodgkin's lymphoma. He came to us and said, I want my own babies, non-obstructive azoospermia. We did a micro TSA. After about 1.5 hours, we found 3 sperms. 3 oocytes were exceed. We got embryos, transferred 2 embryos, which we finally got, and delivered 2 twins. But that was a day when the father came to us, and you know what he said? He said that my family, Babaji, had told me that my child would survive, and I took him to Mumbai to Dr. Advani. He survived. And the Babaji told him, there is somebody who would give you your own children. And I reached here, and I have my own children. The truth is, that moment gave us so much happiness, confidence, and apart from that, that feeling of something which is priceless, otherwise as everything else MasterCard can buy. That was the time from 2014 that we started doing micro TSA. So in non-obstructive azoospermia, it's like a tree with apples, and you've got to find these apples in the testicles. You could do a single, you could do a window technique, which means a simple TSA first. And in this simple TSA, you could take a small sample and from a small window, pick up a chunk of tissue. This normally would be done in hypospermatogenesis as the first thing which we would like to do, because in a hypospermatogenesis, the chances are we could even get sperms in this. So why open up further? Now, a multiple incision TSA is something which we used to do earlier before we used to do a micro TSA. And in this, you would normally open up the testes. This was a patient of maturation arrest who had a biopsy earlier, so the tunica is stuck to the rest of the testes. So after opening up the testes, we would like to take multiple biopsies, and from these multiple biopsies from different areas, see if sperms are there. So this would be a multiple biopsy TSA. This is how a TSA sample would appear to be. And we normally strip the seminiferous tubules. And once they have been stripped and the homogenate has sperms, we would normally observe the homogenate and look for any sperms which are present so that we could use them, and if they're extra, so that we could freeze them. So this is a painstaking job, and let me be honest with you, there is no surgeon who can be successful without a team of embryologists. So what you need is 2 things. Number 1, patients, and number 2, patience. And these 2 things ultimately give you the mastery of sperm retrieval, because the retrieval is not done in the testes only. The retrieval is done from the specimens which you send. And hats off to all the embryologists who put hours into it with the surgeon. So needle mapping is something I'm going to talk to you about. So needle mapping is a beautiful procedure if you would like to do before you do a micro TSA, because what you actually do is you put in about 16 areas, 8 above the equator, 8 below the equator, and you would do like you would do for a TISA. You would like, the moment the needle comes out, you would see if there is any seminiferous tubule which you're going to pull out. And then you would know exactly from the result if there is an area in which sperms are present. And if they are not adequate, you are going to go into that area to pick up those sperms. So you would have 16 dots and 16 areas you would like to do a needle mapping in. But something which I normally at times do, especially in a hypospermatogenesis, before I do a micro TSA, I do a TISA mapping of the testes. So what I do is that you can see the testes over here. Once again, after taking the testes out, I would mark about 16 areas, 8 above the equator, 8 below the equator. And what will you do over here is, after you've put in a needle and you've given suction, you would take out the needle with suction. And when you do so, you would have a seminiferous tubule, take a microforceps, pull the seminiferous tubule, and since it's a tubule and you keep on pulling it, you get a relatively beautiful piece of tissue from a needle hole. And this you would do at 16 areas. And since it's adequate tissue, normally sperms would either be got in this, or it will give you a fair idea of which is the area in micro TSA that you would be able to get a specimen from. So this is what I normally have started doing before a micro TSA. But a micro TSA is a beautiful procedure. Let me be honest, it's magic. And I never knew that my training in andrology in the 90s, I mean 89 to 93, and doing microsurgery at that time, which I really did not like at that time because there was very little blood in it, would be so fruitful to finally give a children to so many. So what you normally would do is do a magnification of 16 to 25. And during this magnification, see if there are any enlarged seminiferous tubules. So if any enlarged seminiferous tubules are there, you would then consider that they could be having spermatogenesis. So you're going to take out these enlarged. In case you do not find any enlarged, you would look for seminiferous tubules which appear to be different. And if the tubules are all identical, and before that, of course, you've got to see the blood supply. So normally where the blood supply is not too much is an area where you can get these tubules. And more importantly, if all the tubules are identical, then you could take random biopsies. 3 from each pole is what normally is done. And this is very useful and very helpful. So this is how a micro TSA would be done. So what we would do is, this is the first step. The moment you open the testes, you would not bivalve the testes initially. You would like to take out tissue from the non-bivalved area, especially an area where you feel there is a different kind of a tubule or an enlarged tubule. And the embryologist, of course, is simultaneously going ahead and stripping them. So you would open it up, send the specimen, ask the embryologist. If the embryologist says, I found, you call it a day. If the embryologist says they have not found, then you'll go into the second layer of the testes. Now, this is how, once you have bivalved the whole testes, opened up nearly the whole testes in different planes, you would go into different areas. And from these different areas, you're going to take out small specimens. The testes is beautiful because even if you open up the whole testes, it ultimately closes back like normal. So different specimens are taken from different areas into Eppendorf tubes, and this is then sent to the laboratory. Now, this is how it would appear to be an area of enlarged tubules, which will be picked up and sent to the laboratory. Now, in a testicular sample, once you have stripped it, cleared it with a lot of debris, this is how, after stripping, you would find in an area a sperm. You have this mobile sperm over here, which is moving the whole specimen, and that's beautiful. And this is how you would find these sperms and pick them up. Now, how do you process? So the procedure is followed as the same for TISA. Testicular fragments are washed so as to remove blood clots, mechanically dispersed using tubercular syringe until no intact tubule is found. Now, this raw suspension could be concentrated, it could be washed, it could be resuspended, a density gradient could be done, and we normally leave it to be incubated for some time so that the sperms could gain some motility. So there are different strategies to identify live immotile spermatozoa or dead ones. You could do a Haas test, but you could also do what we normally do, a sperm flexibility test. You touch the sperm tail and force the tail up and down, and flexibility is a marker of viability. We also use pentoxifylline, and now there is a new media which has come up with theophylline. I mean, that's a beautiful media to be used in these specimens. And once again, you prepare this plate depending on what you think you will do and how you would do it. So the embryology and the embryologist is just like a surgeon. He prepares his mind where to open, how to open, what to take out, how to take back, what to put back. And similarly, a dish could be made where you could have a large spread because you feel you would take time to get the sperms and place them into the drops. Or you could have a large spread with 2 drops from where to transfer them into the PVP and immobilize them and to be used. So it's a beautiful science for the embryologist as well as the surgeon. In fact, from some of the best friends which you could have are the embryologists, because you share the joy together. A testicular sperm extraction, once it is done, this is a very old video. I mean, I think it's a 14-year-old video which I had made, which I've just placed over here because I thought it will be appropriate. So you would pick up a sperm. The beauty is that once you have got sperms, they could, if the shape is better, good. If the shape is not too good, even then the genetic material could be fine to have a baby. And just to go ahead and do ICSI with these sperms. So a few things which, once you have taken out the sperm, the extraction has been done. There are different ways of handling. You could do, of course, micro TSA, you have a very little specimen, so it's optimal tissue to increase the efficiency at times. But at times, of course, it's very taxing too. So you could do mechanical mincing, you could do enzymatic mincing, erythrocyte lysis, which you always need to do in case it is there. What we normally do is just wash them with the sperm medium. Motility enhancement is needed with pentoxifylline or theophylline. You need to be very, very clear about the quality of your lab, the temperature stability, the centrifuges, and the sterility of the equipment. And of course, ICSI is used, Haas test may be used, and please have a good program of sperm freezing. Even vitrification you may need in case you get these sperms, because they are precious. And doing an operation again is quite taxing on the patient. So you could do swim up, density gradient, subjectively sperm selection. Dr. Vasan has spoken in detail about it, about IMSI, PIXI, electrophoresis, bifringence, microfluidics. I won't talk much about it, but all that I like to say about the advances is that they have not been considering the cumulative success rates. We do not have too many statistics to detect small differences. The lack of independent studies, there's poor and low quality evidence, lack of generalizability, they're expensive. So of course, they're very good. And the particular case of ART in medicine results in a combination of two individuals and three components, sperm, oocyte, and endometrium. And whatever new comes, we need to use it and then think about it, what is actually useful to our patients in the long run. Sperm freezing, of course, is the most important thing over here. It could be done in low volume carriers. And the take home message is that non-obstructive azoospermia is beautiful in severe male infertility conditions, I mean the microTC. And despite lacking sperm in the ejaculate, 50% you could have their sperms. You need to do a few things like counseling, genetic diagnosis, identification of men who are ineligible, use of microTC, application of the correct techniques. You need a beautiful multidisciplinary effort between the andrologist, geneticist, reproductive endocrinologist. You need an embryologist who loves this work. And you need to space your cases so that even if this patient takes 2 hours, the rest of the patients are not in a hurry. And stem cell technology is going to be beautiful in the future. I'll just talk in 4 slides about it, and then I'll call it a day. In the future, you could have the tissue for a xenotransplant, organ culture, or autotransplant. But even a pluripotent spermatogonial stem cells could be transplanted or cultured to get a sperm. But a pluripotent stem cell could be used to have a sperm in the future. So spermatogonial stem cells in non-obstructive azoospermia, where you have not been able to retrieve cells, could be a beautiful modality in the future. In mice, in non-obstructive azoospermia, spermatogonial stem cells have been cultured to get sperms. These sperms have been used to microinseminate mice, and babies have been born in mice. It has not been possible in humans till now, but it will be possible one day or the other. CRISPR-Cas9, another beautiful technology, which will be used in the future on embryos to have babies free of disease. But the most beautiful thing which I'd like you to think about, I have got this slide a lot of times because it really runs my mind. In the future, a man and a woman have a sperm and oocyte and have a baby. 2 men get married. Till now, one gives a sperm, the other takes a donor egg, is put in a surrogate, but a pluripotent stem cell could be used to make an ova. And with that sperm, both could probably have their babies in a surrogate. And in the future, when 2 women want to have a child, you could have a sperm from a pluripotent stem cell of a woman and an ova from one, and it could be placed into one of them to have their own babies. But they would never be able to have a son. And I end at this note. Thank you very much for your kind attention. Love to be here with you. Thank you.
Thank you, Dr. Watson, for the wonderful talk, and it was really interesting to know a lot about that. And you made it sound really like, I remember when we used to learn surgical retrieval, it was found like very difficult in an embryology lab and get the tubules, and you have to do the scrapings and find the sperm. When you find a single sperm, it makes you so happy. And
oh, yes,
as a gynecologist, that, oh, we got it. Great. So tech team, we can move ahead with the next talk.
All right. Before we go ahead, I want to say this. Dr. Jindal is an orator.
Yes.
सर से मैं बड़ा प्रभावित रहता हूँ.
No, it is your kindness, and thank you all of you for inviting me.
And with the habit of sir that he connects to the with the soul and the heart of the listeners now with his words and his slides. Thank you so much, sir, for joining us.
Thank you very much. Thank you.
Can we have the next slide, Vakil? So the next speaker is our second coordinator, Vakil Jagveer. He is actually doing his PhD from Lovely Professional University. And he has done his embryology training from Biomedical Research, Chennai, associated with IIT Madras. And he's a life member of IFS, ISAR, NACHI. And he trained people on semen analysis, sperm morphology, sperm DNA fragmentation, advanced sperm selection techniques like microfluidics, MAX,
And currently he's working with Cooper Surgicals as a clinical embryologist. Welcome to you.
Thank you, Gaurav. So I'll share my screen. So tech team, I'm not able to share. Yeah, now. Thank you. Just let me know when it's visible. I hope it's visible to all.
Yes, sir.
Thank you. Thank you so much. So we'll discuss the effect of high leukocytes and DNA fragmentation in male infertility. And in the previous talk, I think this topic has been a little bit discussed. Dr. Vasan has given us a beautiful insights on how to do DNA fragmentation testing with the latest methods available in the market, and also the effect of ROS and all the things we have discussed. So we'll have a brief insight on the same from another point of view. So leukocytospermia, we all know when we have leukocytes or WBCs in the semen sample, more than 1 million per ml sample is considered leukocytospermia. So it may come from many reasons, and the thing which is a troublesome thing is that leukocytes are found more in fertile men than in fertile men. So that is something which we have to consider. So the causes of leukocytospermia could be infections, it could be venereal disease, it could be the routine infections of urinary tract or the reproductive tract. It could happen due to inflammations, it could happen due to autoimmune conditions. And in varicocele, we have already discussed in the previous talk that varicocele could be a cause of leukocytospermia in males due to some systemic and prolonged illness and infrequent ejaculations. Like if a person is not ejaculator for the longer duration, you may find more WBCs, more leukocytes in the semen sample. So if you see, the infections in the genitourinary tract may play a contributing role up to 15% in male infertility. It's a well-known indicator of infection and inflammation, which we all know. But the trick here, or the twist here, is when you start with a diagnosis of this. So first of all, initially for the diagnosis of leukocytospermia, we usually do the wet mount. From wet mount, we found to know that this person has more WBCs in the semen sample. But the issue with wet mount, if you are a consultant and you're going to semen sensitivity or culture for bacteriological investigation of the semen, just on the basis of wet mount, where you see more round cells or cells shaped round and have some granulated morphology in a wet mount, that may not or may be the WBCs. There may be immature spermatids as well. So to rule out that, we from wet mount we move to the peroxidase staining using orthotolidine, where our neutrophils, our leukocytes, they become brownish in color. Then you can assess them that these are, yes, what I am seeing, round shape and granulated, are not leukocytes, or they are leukocytes. If they are leukocytes, they will be brown in color. If they are not, they will be transparent. There may be immature spermatids or some other round cells. Then you have no need to send it for culture sensitivity. Then you may have no need to put in on the antibiotic as well, because being a member of emerging antimicrobial resistance society, I find it very odd when we put a patient on antibiotic, because antibiotic may have adverse effect on the semen parameter as well. Then apart from the routine peroxidase staining, you can go to the next level where you can do the staining for CD45 marker with immunocytochemical staining, where your leukocytes will be perfectly stained in a dark red color. You can see that actually what we are seeing in the semen are leukocytes or immature spermatids. But after that, when you have the confirmation that my patient has this leukocytospermia, then you can send it for bacteriological examination, where to find out the cause of leukocytospermia is to do with pathogen or which bacterial microorganism. So what is the impact of high leukocyte on male fertility, which we already learned in the previous lectures, that having high seminal leukocytes will lead to reactive oxidation and high oxidative stress, which can impair sperm motility, sperm morphology, and which can also lead to sperm DNA fragmentation, and which we have already discussed. So it is, we can say it's a complete oxidative attack when you have high number of leukocytes in the semen sample, which are not good for your routine sperm parameters. So overall impact of leukocytospermia and male fertility, if you see the relation between leukocytospermia and sperm fertility remains controversial. Reason being because we are not diagnosing it properly. That's my personal opinion, that sometimes we may find immature spermatids and we label them as leukocytes, and then we send it for cultural sensitivity. We found nothing. There's no impact. There was no impairment in the motility or the impairment in the DNA of the sperm, and we say that okay, this sample has had leukocytes, but there was no impact on the male fertility potential. But that may be because of the incorrect diagnosis, I would say. Then, but the studies which says that yes, it can impair the spermatogenesis by altering the cytokine levels, and which can impair the Stoley cell functions as well. And it is found in teratosthenospermic samples, and necrospermia is found more in leukocytospermic men than in normospermic men. So there's a correlation which we have in the studies. A meta-analysis which evaluated the impact of leukocytosperm in men attending a fertility clinic found no association between condition and the reduced fertility after ART and with altered semen quality. So that's what I said. It is the correlation between them is still not much established, or we can say still controversial. That some studies says that yes, it has the impact, and some studies say the otherwise. But apart from that, if we're putting the patient on antibiotic therapy without ruling out that the person has leukocytospermia or not, that antibiotic therapy can impact your sperm parameters. Even there are studies who states even the patients with leukocytospermia, when they were put on antibiotic therapy, that number of leukocytes were reduced, the infection was ruled out, but there was a significant impact on the sperm parameters like motility and sperm DNA quality. So next, we'll move to the next part of our presentation, which is DNA fragmentation. Apart from leukocytospermia, that was all, which I think sufficient amount, and it should be a routine part of your semen analysis. The leukocyte counting should be there in your routine semen analysis, that you should know that the patient has leukocytospermia or not. On the other hand, DNA fragmentation, which has a major role, which is in the current, I will say currently hot topic when we are discussing male fertility potential. So we all know what DNA fragmentation is. That there are fragments, there are nicks on the DNA where some base pairs are missing, and those base pair may be responsible for some significant amount of protein, which are responsible for some significant genes, and that may lead to failure of your cycle, that may lead to congenital deformities, that may lead to adverse effects in the point of view of ART outcome. So what are the causes of DNA fragmentation? So initially in sperm, if we study the spermatogenesis thoroughly, we found that some of the sperm abnormalities either is related to its DNA, or is related to its morphology, or is related to its vitality, are originated from its origin itself. Like when the sperm moves from mitosis to meiosis 1, from meiosis 1 to meiosis 2, from the spermiogenesis, spermiation, and that comes as ejaculate to us, there are some abnormalities which occur at each step. Like for the first, if we rule out the cause of DNA fragmentation, the induction of apoptosis at the point when it moves from mitosis to meiosis 1. So the nature induces, Stoley cells induces apoptosis on the cells where the genetic material is not properly confined. Those cells goes under apoptosis. But this process occur up to 80%, I'll say, just a random number I'm saying, doesn't occur 100% foolproof. For some of the sperm with DNA fragmentation move to the next stage also. Then at the time of spermiation, when a sperm becomes flagellar from a spermatid, from a round spermatid, at that point of also, it will have some impact on its DNA, and as well as its morphology. When a large cytoplasmic droplet remains intact, its neck, which we all know that cytoplasmic droplet on the neck from where it comes. Then the time of spermiation, when the sperm travels together for seminiferous tubules to epididymis, and they stay in epididymis in the sperm with fragmented DNA, and the impact of ROS damage the other sperm which does not have the DNA fragmentation. Then the activation of endogenous nucleases and capsids due to scrotal temperature changes, or due to more amount of time they're staying in the epididymis with other infected cells. Then due to radiotherapy, chemotherapy, and then there are extrinsic factor also, which are more lifestyle related and physiology related, like varicocele or some infections. So diagnosis, we have already seen in the lectures of Dr. Vasan that we have various methods to diagnose DNA fragmentation. So I will cover mostly the methods from automated to the manual, which Dr. Vasan also already covered. So methods to diagnose DNA fragmentation in sperm, there are various methods available in the market, starting from the automated, completely automated, electrophoresis based, fluorescence based. Then we have manual methods like alanine blue, and then we have sperm chromatin dispersion assay, which we cover. So each method to evaluate sperm DNA fragmentation vary greatly in both method and type of damage they are detecting. Like tunnel assay and comet directly assess the presence of single and double strand breaks. On the other hand, SCS and SCD detects susceptibility of chromatin to treatment by the acid. So tunnel is basically terminal deoxynucleotide transferase nick and labeling, where we use fluorescence microscopy and fluorescence dyes. So the degree of fluorescence, when the degree of fluorescence increases, it shows that the number of strand breaks are more in that particular sperm. Then comet assay, where we use electrophoresis. The bigger the comet tail will be, the more fragmentation is there in the particular cell, the particular sperm. Then we have sperm chromatin structure assay, which we already saw, seen a video demonstration as well from Dr. Vasan, where we see both the single and double strand denaturation. And then we have the most common and most widely used method is the halo method, sperm chromatin dispersion assay, where we do use a routine bright field microscopy and we treat the sperm sample with acid, and then we lyse the sperm head and we stain it. We see this method is performed widely because it's a manual method. The kits are ready to use available in the market. It can be performed within an hour and there's no equipment required for that. So if we compare all the methods which are commercially available or which I'll say been tried and tested, Comet, Tunnel, SN, SCD, so all have the pros and cons. But if we compare from the Comet Tunnel SSA with SCD, okay, it is cost effective and there's no equipment cost is there, but the sensitivity and specificity of this method is still under questionable, and still intra observer variability will be there, because one person may see that halo, because there are different issues that you may assess the halo as large halo or medium halo or small halo. Sometime because of the mishandling by the user of the kits, you may not see halo, you may see all halos, they miss some steps. So there are chances where you make more errors. And apart from the other methods, okay, they are a little bit expensive than this, but they are tried and tested. Their sensitivity, their specificities are properly tested. They have well standardized procedures. They have well standardized CV percentages. We know they have quality control methods. So depending upon your, I'll say your comfort or your affordability, or whatever you can say it, you can choose the method, then you can apply it. The more manpower you can spare for the particular test, because this test takes time, even if you're doing with a manual method or with the automated method. Then the impact of DNA fragmentation on the reproductive outcome. So I'll start from the natural pregnancy, what the study says, then to IOI, then IVF and ICSI, in all three methods of reproductive outcome, and what are the studies will say. So in a natural pregnancy, there was a meta-analysis involving 3 studies of 616 couples. So that high DNA fragmentation determined by SCSA was associated with failure to achieve natural pregnancy with an odd ratio of 7.01. And in a fertility environment study where around 500 couples with no infertility history, SDA was found to be associated with the cause of delay in conception. So sperm DNA fragmentation impairs even in a natural pregnancy, because the factors affecting the sperm DNA are intrinsic as well as extrinsic. So the environment contaminants, your lifestyle factors, as we discussed, all those factors, they can impact even a fertile person, okay, who is not seeking the infertility treatment, they can also get impacted with the same. Then in IUI, the association between SDF and poor IUI outcome is not without debate, because in a study, SDF more than 30% was a predictor of decreased pregnancy and delivery rates after IUI. And in insemination of more than 12%, tunnel positive spermatozoa resulted in no pregnancy in another study. And research study also that SDF index of more than 27% by SCSA has a negative impact on IUI pregnancy rate. So if I'm thinking that I should do DNA fragmentation testing only in the patient who are seeking treatment for IVF or ICSI, no, I should. If I feel that if I suspect that the patient may have DNA fragmentation, and I think for the routine testing, I will not recommend it for the routine testing. But if the person comes to you for seeking infertility treatment, I think this test with the method which is affordable to them as well should be implemented in the infertility clinics. Then for IVF and ICSI, the outcome is different in IVF and ICSI, as IVF, both the gametes are subjected to prolonged culture, and we know that oocyte still has the ability to repair itself. And therefore, the fertility rate of IVF may be affected when there is a larger population of sperm or with high DFI. But on the other hand, ICSI, the better quality oocyte with the less exposure to oxidative stress may be better to preserve its ability in repairing SDF to a certain extent. So that's why when we see that, we know it's a well-known fact that ICSI could be a good method in a patient with high DNA fragmentation index. So there were lower pregnancy rates seen in patients with high SDF was observed by evaluating 9 IVF studies. systematic review failed to find a significant association between SDF and ICSI pregnancy rates. But a meta-analysis with 16 cohort studies, including more than 3,000 patients, reported a lower pregnancy rate in context of high sperm DNA fragmentation only in patients undergone IVF, but not in ICSI, because in ICSI, somewhat you do the sperm selection, like you try to select a good morphology and good motility, and the evidence-based study says that a sperm with the high progressive motility tends to have a chance that they will have low DNA fragmentation. And in ICSI, we try to select those sperms who have a better quality with respect to morphology and with respect to motility as well. So on the other hand side, the oocyte also has the repair power, regeneration, repair power. So they somehow manage things. Then a recent meta-analysis includes 56 studies in which they concluded that SDF predicts a poor clinical pregnancy rates for both IVF and ICSI. So studies are still going on. Gaurav had some papers on the same. He presented in ASHRAE where he compared high DNA fragmentation patient and he applied multiple methods of sperm selection and found significant and good results with that. So this is a beautiful paper which I wanted to highlight because usually we believe that if a sperm has high DNA damage, or sperm has a DNA damage, basically a semen sample has a high DNA damage, in those patients, I may not have good fertilization rate, I may not have like good embryo quality. But in this paper in Asian Journal of Andrology by an Asian group, they did around a good number of cases of ICSI and IVF combined, and they divided those patients into 3 groups with respect to their sperm DNA damage, less than 10% sperm DNA fragmentation, 11 to 20%, and more than 21%. So if you see, fertilization rate is similar in all the 3 groups. There was not huge impact on the fertilization rate when the patient has even low DNA fragmentation index in the semen sample, even if it's a high DNA fragmentation in the semen sample. But by the time it reaches day 5, the blastocyst formation rate was less in high DNA fragmentation patient pool group, and the pregnancy rate was pretty much lower and significantly lower in the makeup, the pool of high DNA fragmentation patient, and simultaneously the abortion rate was more. So if we conclude with this study, that it just gives an insight that it's not necessary that your sperm DNA fragmentation will impact your IVF fertilization or ICSI fertilization rates. It will going to impact post that. So what are the treatment options we have for high DNA DNA fragmentation? So for conventional, we have short abstinence method, which we usually apply when we are doing semen analysis as well. Or we have patients with high DNA fragmentation. We have to rule out that this patient particularly have high DNA fragmentation, or it could be a one-time thing because due to stress or due to some abnormal conditions, they may have high DFI in one sample if I'm using routine methods like Hello method or the basic staining methods. So it was found that 20 to 25% reduction was achieved with ejaculatory abstinence of 1 to 2 days without compromising conventional semen parameters. So it could be a good thing, like you have a shorter abstinence sample in couples with high DNA fragmentation if you're not using any other methods or other sperm selection techniques, which we're going to discuss in further slides. Or you can try an oral antioxidant therapy, which I think most of the clinics there nowadays are trying because they are finding couple with men with high DNA fragmentation. So the studies suggest that yes, they help at some extent in couples in men with high DNA fragmentation and in reducing the DNA fragmentation index. But still, we need more studies on that. Still, more data is required. Varicocelectomy, we already know post varicocele repair. There was a systematic review, including 511 patients, showed highest level of SDF in men with varicocele and improvement in the DNA fragmentation index after varicocele repair. So then the another method, which we can, which I'll call a quick method of sperm selection, that while doing ART procedure, if you're not wanna spend some time in and wait for oral antioxidant therapy, or if you don't get good amount of sperm, even after short abstinence method, you can try for sperm selection. Whereas there are various methods available in the market, and as Dr. Vasan suggested, that yes, there are methods in the market, but still we need more data, still we need more research on those methods. Yes, the results are not good, or neither good nor bad. We are getting, some studies are saying yes, methods are really helpful, some studies are saying yes, there was no significant improvement, but still with more research we may have to find more data. So there are various methods, like there are hyaluron based sperm selection methods, PIXI or sperm flow. PIXI is basically physiological ICSI. Then we have magnetic activating cell sorting. Then we have intracytoplasmic morphologically selected sperm injection, IMSI. We have microfluidics, or I think testicular sperm if you don't want to use any other method. But for testicular sperm, you need to have a person like Dr. Jindal with you. So sperm selection methods, will start with the hyaluron-based sperm selection methods, ICSI and sperm source. It's basically based on the principle that the mature sperm, mature in the sense of sperm with high DNA, like mature sperm, mature with respect to the DNA integrity, have affinity toward hyaluronan or hyaluronic acid, which is naturally present in the cumulus. So this is one of the part where sperm goes toward the cumulus and attached to it. Okay, it's believed that the sperm which has ability to bind, it's not believed, I'll say it is found out with the research that the sperm's ability to bind with hyaluronic acid correlates with maturity, morphology, and high DNA integrity. So with that method, in vitro techniques were designed like PIXI, physiological ICSI, and sperm slow. In PIXI dish, we have basically dry hyaluron dots, which are hydrated using your buffer media, and we put the semen sample. The sperm, which are mature with respect to hyaluron receptors on them, we stick to those hyaluron dots. We pick those sperms and we use those sperm for ICSI. So what are the studies say us? So studies suggest us that use of hyaluron-based sperm selection for ICSI is reported to reduce the proportion of pregnancies that end in miscarriages. And also there was a study by Dr. Agarwal's group. The study shows that there was an improvement in live birth rate with PIXI against the control group where no intervention was used to reduce STF. Then we have MAX, which is magnetic activity cell sorting. Dr. Vasan already explained about a little bit about this. So it's where we use paramagnetic microbeads coated with an Nexin 5 protein. So apoptotic sperms has affinity. Apoptotic sperms have phosphatidylserine, a lipid on the head, and an Nexin 5 has affinity to bind that lipid. And the Nexin 5 is coated on a paramagnetic beads. So the sperms which are apoptotic in nature, they will bind with the beads which are labeled with an Nexin 5. And then that suspension will be passed through a magnetic column and they will stick to that magnetic column. And sperm without apoptotic markers, they will fall in the container which is kept below. So what does the data say? So MAX shows potential to optimize SDF reduction rate and applied to raw semen before density gradient centrifugation and swim up, especially in samples with low values of progressive motility, vitality, and hyposmotic swelling tests. It helps in reducing the sperm DNA fragmentation. And the use of MAX in patients' altered levels of DNA damage seems to improve the sperm quality. Still, we are looking for more studies with respect to ART outcome. Then we have IMSI, intracytoplasmic morphologically sperm selection. So in IMSI, basically we use a high magnification lens, which gives us a magnification of 6300 per x. So it gives you better morphological selection of sperm. So you can see sperm morphology in a better way. You will be able to see the vacuoles. You will see a sperm head in a better way. So better sperm morphology plus better motility will have a better sperm quality with respect to a raw semen. So patient with high sperm DNA fragmentation rate, selection of normal spermatozoa with vacuole-free head yields in greatest likelihood of obtaining spermatozoa with non-fragmented DNA. And using IMSI showed modest improvement in live birth rates in high SDF cases as compared to control group when no intervention was used. So it is also one of the useful method which you can do, but still we need more data on that. Similarly, as for the microfluidics, where we use sperm's ability to move in microchannels, a good method, beautiful method where you avoid centrifugation completely, because centrifugation is considered as one of the thing which can induce ROS and which can lead to DNA fragmentation. It shows the technique is relatively new, okay, but still the studies show that it helps in reducing the sperm DNA fragmentation with respect to density gradient centrifugation, because in density gradient centrifugation is involved. There are very few studies improving the showing in the reproductive outcome, but the studies are there which shows that the DNA fragmentation is reduced when microfluidic is used, but not very much studies in respect to reproductive outcome. Then I think this could be a good thing if we do this patient's own sperm, and not that much of outside interventions used, but still you need to have a skilled person who gives you a good testicular sperm, good testicular biopsy or testicular extraction, and then you have to be a skilled embryologist to extract sperm from that particular sample. So the 3 to 5-fold lower sperm DNA fragmentation observed in testicular than ejaculated sperm support the use of testicular sperm in ICSI as a treatment strategy for high sperm DNA fragmentation. Studies have shown promising results regarding the use of testicular sperm and ICSI in men with high SPF and oligospermium, which it counts as both. Even if you have less number of sperm, you can go for testicular sperm. You'll also found like Sone Pe Suhaga, sperm number was less. You went for testicular sperm and you found sperm with high DNA integrity. The use of testicular sperm in ICSI in couple with previous ART failures are further supported by emerging data, and a significantly lower tunnel positive rates in testicular sperm and a 50% pregnancy rate were reported with the use of testicular sperm in subsequent ICSI. So this can be a good method if you have the facilities to do this. So to summarize this, we all know that semen analysis is the cornerstone of male fertility diagnosis, but a good semen and a thorough semen analysis, which gives you a good insight about the patient's sperm parameters, should be must. So it helps you in planning that what will be my treatment option for this particular patient. Like if you have high leukocytospermia, but you are not able to diagnose it, you are considering it as immature spermatids, or if you have high number of immature cells, but you are labeling them as leukocytes and you're putting the patient on antibiotic, that may impair other sperm factors. So you have to have a thorough diagnosis, and leukocytospermia diagnosis, which can be made part of a routine semen analysis. DNA fragmentation we can think about using for each patient, but if you want, you can think about patient's previous history. If they're having previous ART failures and you are suspecting a male factor infertility, you can go for DNA fragmentation testing for those particular patients. Leukocytospermia diagnosis is simple and can save time, cost, and unnecessary use of antibiotic, which is very much necessary, not from a fertility point of view, from an immunity point of view also, because unnecessary use of antibiotic is not good for any human being. Sperm DNA fragmentation testing methods can be chosen on the basis of its suitability to individual clinic, like whatever suits you, you can go with that because it gives you an initial idea. But if you are using the methods which are manual based or which are not having strong data backup, I would suggest you to go for a sperm selection method as well to improve your outcome. Then sperm selection high STF patients can help in improving the ART outcome. Similarly, in that also, you should choose a method according to your suitability, according to the cost effectiveness of the method, according to the data and the studies research supports that method. So that's all from my side. I hope I finished within the time. So over to you,
Thank you, Vakil, for the very nice talk. We all enjoyed your presentation. So thank you, Vakil.
Thank you.
Can we move to the next speaker, please?
Yes. Tech team.
Anshu. So next speaker is Gloria Martinez, and she is a very renowned biologist. She is from Microoptics, Barcelona, Spain.
Hello, today I'm going to talk about quality control during semen analysis. So, first of all, what is quality control? Well, it could be said that quality control is a combination of all of the measures to ensure the early detection of measurement errors and the procedures to rectify them. These steps need to be done in order to obtain reliable and accurate tests, and of course provide the patient with the best possible patient care. If we had absence, if there was lack of this quality control, this could lead to a misdiagnosis, incorrect treatment, and finally increased costs to the hospital or the clinic, the laboratory. So some of the reasons why we need quality control are the increasing necessity to get reliable results for getting proper diagnostics, as I have said. Also, there is an increasing importance of detecting and correcting systematic errors and significant deviations during sample evaluation. Of course, it will be very different if these errors that we can appreciate during semen analysis are due to the counting chamber, that it has some problem, or some problem during inter-technician variation. So it's very important to identify the origin of the problems. Of course, also there is a large number of discrepancies between specialists, and this can produce subjectivity. For example, each technician will have their own criteria, and if they use a CASA system, maybe they can introduce manual modifications. So the procedure to follow needs to be established. Therefore, there is a requirement of a method to evaluate the origins of this result variability. And as Dr. David Maculo said, finally, in brief, quality control brings stability to the laboratory. We are going to try to identify the type of errors that we can run into during semen assessment. On the one hand, there are random errors that we can find. These errors are caused by differences in readings or in sampling. And they are assessed by repeating the measures on the same sample and using the same observer and the same equipment. So in this case, we won't see a tendency, just different results between replicates. This is different, on the other hand, but because we have the systematic errors also called bias. In this case, there is a tendency to give results always too high or too low. This is not detected with repeated measurements, and if we only use a specific technique to do the semen assessment, it's possible that we cannot identify this kind of errors. And these are arising from factors that affect the results in one direction. So it's good to have in mind these two kinds of errors that we can run into during semen analysis. And why? Which are the reasons of these out of control values that we can find? First of all, the problem could be an inadequate mixing of the sample, because as you know, semen samples are heterogeneous, sometimes viscous, and it's very, very difficult to homogenize well these samples. Also, concerning technician stress, of course, at first hour in the morning and at the last hour of the day, the technician stress or the technician work will be different. So we need to keep in mind this. Also, maybe there has been a poor technique during pipetting or simply handling the sample. Or maybe there is an inadequate training to the technicians. The training is very important, and the intention is that more or less everyone has the same criteria, at least in a clinic, in order to have an homogeneous opinion and an homogeneous way of working during the semen assessment. Also, maybe there is instrument variation, so it's very important to make sure that everything is calibrated, that the microscope has all of the settings correct, the phase contrast, if you work with phase contrast, that they are well aligned, also the balances, so all of the equipment in the laboratory. And finally, another reason can be the deterioration of quality control samples. Having said that, now what we can do in order to minimize these sampling errors? Well, in this case, the first thing to do is maybe to use the positive displacement pipettes, which are more accurate, and with them we can obtain correct ejaculate aliquot volume. Of course, we cannot forget the dilutions that we need to do in our everyday working in the laboratory, in andrology laboratory. So it's very important to perform well the dilutions when using the counting chambers. For sperm concentration and motility assessments, it's recommended to do replicate assessments. This has been maintained in the new manual of WHO 6th edition. Also to use a sufficient depth of wet preparation for sperm motility, so that the sperms have enough space to swim, and then the motility that we are assessing during the analysis is the correct one, the real one. And finally, importance of number of sperms assessed. In the last manual, we keep with 400 sperms for motility and concentration analysis, and it's recommended to do replicates, and 200 sperms for vitality and morphology. In theory, in this edition, it's not mandatory to do replicates for vitality and morphology, but it's good to have a second slide in case there is some kind of problem with the first one. Next, which types of quality control are there? We can differentiate basically two kinds of quality control. On the one hand, the internal quality control and the external quality control. In the next slide, we are going to go deeper in which concerning the frequency to do this analysis. So the internal quality control, it's basically the use of test controls like standards to validate the testing process and the equipment. So here what we are checking is that the systems, the equipment that we are using, work correctly, so that the results that we obtain, they are reliable. This can be useful in order to test the intra-technician variability. So one technician repeated measures to test the equipment. So the recommended frequency can be different for different centers. If one center analyzes a lot of samples every day, they can do this every day. If not, if they have less samples, maybe every week and so. But of course, each center will set their own protocol and they will have the frequency of quality controls set. In contrast, the external quality control, here we need to think that, well, we give as said that the equipment is working well, and that in this case we are comparing different technicians, so between technician variability in order to test their criteria. In this case, there are different cases of external quality control, and there are some EQA programs, external quality assurance programs, provided by different companies. After also we are going to talk about this. But the most important differences is that in external quality control we are taking for granted that equipment work correctly, and then that the criteria between technicians is being studied. So this is a slide directly from the WHO 6th edition manual in order to study a little bit the time schedule set for quality control. So as you see, there are different procedures to run. Some of them all of the times, others just weekly, monthly, quarterly, or biannually, or annually, for example. So it's important to take this into account. In this slide, I wanted to share with you some differences that can appear during semen assessment. As you say, there are different techniques that can be used for semen assessment. Some of them are manual techniques, and others count on CASA systems, which are more automatic. So there are a lot of centers that work with manual technique, others that work with automatic CASA systems. So in my case, as I am part of Microptic developing CASA systems, I will explain a little bit the difference between both techniques. Just to summarize a little bit, it could be said that a manual technique can be more subjective, because at the end each technician has a little bit their own criteria. They could be very similar indeed, but it's true that a lot of training is required, a lot of time also, and it can be of course more time consuming, and there is therefore lack of traceability, because it's not possible to have access to the analysis that the technician has performed. What the technician sees through the microscope are the results that they will write in the final report, but it's not possible to have the images or the videos or more calculations. So in contrast, when using a CASA system, there is a gain of objectivity, because each analysis is done always like the same. So the results that we obtain are more reliable, more consistent, and faster. And of course there is a gain of standardization for the laboratory, which could give it more prestige. and we are leaving aside the technician variation, different technician variation, and finally also it's user friendly and we can get full reports and save all of the images and graphics that are provided by the software, by the CASA system. So I am going to talk a little bit about these two CASA systems that we develop, which is the SCA Evolution, our open system, and the SCA Scope, our closed system. But of course, in both techniques, no matter if we are using manual technique or automatic technique, quality control is always required. So which are the types of quality control that are compatible with our specific software, which is SCA Evolution or SCA Scope? In this case, you can see the summary of both internal quality control and external quality control. In this case, for internal quality control, we have available the quality control, the QC bits, which can be used for the study of concentration calculation, and also they are very close, very related to the disposable used, because concentration can vary a little bit depending on the disposable, so QC bits can be used to standardize this concentration calculation. Also, there is a micrometer slide, calibration slide, for assessing the distances measurements. It's very important because all of the sperm speeds will be, different speeds will be calculated during the motility analysis. So it's very important to know that the distances that we obtain from the program are well calculated. Also, there are some QC slides that can be prepared and then passed different times in order to check that they always give more or less the same result, and some images stored, which is the same idea. Secondly, there is quality control that MicroOptic creates. You will see later in some other slides, which is taking for granted that the CASA system is working well to assess the variability between technicians. And finally, the external quality control program provided by some companies, some associations that create different supplies, slides, or fixed samples in order also to assess technician variability. So the first one is QC bits. QC bits solution. In fact, there are two bottles containing different solutions of latex bits in suspension. So we can use these two bottles to perform this quality control because we already know the results of the concentration that are set by the manufacturer. So it's required to load them in a counting chamber as if they were a normal sample, and then check that the results are inside the acceptable ranges. So this is for the verification of sperm concentration and homogeneity of consumables and the disposables. And in this case, it's very important, for example, to have in mind that if we are using, for example, a Mackler counting chamber, that each year this counting chamber can be changed, because if not, sometimes when we use reusable counting chambers, the Mackler counting chambers can be worn out, for example. After, we can obtain a report of Levy-Jennings type, and this is compatible with our both systems, open and closed system. Next, we can perform also internal quality control with a micrometer, which is also called calibration slide, and knowing that between long lines inside the micrometer, the distance is 100 micrometers, we can use this information to overlap this micrometer with a grid inside motility and concentration analysis module and the other ones, and test if distances are correct. So here what we are testing, what we are checking is the calibration of the system. The calibration will be the equivalence between the pixels in the screen and the real distances. If we get a wrong result, then the system would need to be recalibrated. Also, we have some QC slides, quality control slides, which is this kit that comes always with the system SCA scope, which are 3 already prepared slides from morphology, vitality, and DNA fragmentation that we prepare here. And then every time that an installation is performed, these are also run, the analyses are run, and the results must give between the accepted ranges. If there is some kind of variation, then we can study if all of the analysis conditions are correct and that everything in the device, in the configuration, is correct. In the same way, this time not slides but QC images. In this case, on each analysis module, which can be motility and concentration, vitality, morphology, there are stored images and videos that can be analyzed every day or every week, and we always will expect the same results. So in case that someday the results are changed, this may be related to a change in the analysis parameters. So this is very interesting if different technicians are working with the system and at some point you see some difference. This can mean that the results will be different due to a change in the configuration settings. For example, the classification of sperm into rapid, slow, immotile, or morphology reference values for a normal sperm. The length of the head, the width, the midpiece, for example. And concerning the micro-optic quality control that I just introduced before, as I have said, micro-optic provides users each year with a quality control set of images and videos for 2 analysis modules, motility and concentration and morphology, and they can be downloaded by each user of the system in order to open the sessions, judge if the different images and videos are optimal for the analysis or not. So here we are testing the criteria of the technicians and also the homogeneity of the different technicians' criteria of the same center. And then they will need to fulfill an Excel file and finally send it to us, so we can prepare the certificate and compare the results to the default results. Finally, regarding the external quality control, as I have also said, there are some external agencies and companies that can create external quality assurance programs. There are different in each country, and the supplies that are provided for the centers that are available are in different formats. Some of them are prepared as slides, others are slides that need to be stained, also some samples for testing, video recordings. And here the important thing is to know that there are some requirements that, for example, motility videos need to follow, need to have in order to be compatible with our systems, for example. So here you can check the requirements. And some examples of EQA programs compatible with SCA are these ones. And this is the document that explains, well, the different steps in order, for example, to transform, to convert motility video into a compatible video for the SCA. So finally, in brief, our take-home message of today's chat is that quality control is an essential part during semen analysis. You have seen that there are a lot of consequences if quality control is not followed during the process. Also, that CASA systems provide a gain in objectivity, traceability, and loss of technician variability. So in this case, it's easier to perform quality control and to control the entire process from the moment from when you receive the sample until that you provide the patient with the report. And finally, that micro-optic is in constant development in order to improve quality control so that the analysis provided by each center can be every day more accurate and reliable. So, thank you for your attention.
So thank you, Gloria, for the wonderful talk. So I request the tech team so that we can proceed for the next session. So I would like to introduce the speaker for the next session, which is Gloria again. So Gloria is has master degree in clinical analysis laboratory from Barcelona. She is currently working as biologist in micro-optic, training in courses and webinars, supporting the scientific team, software testing, essay evaluation, essay scope, installations, technical support, and technical documentation collaboration. So I request the tech team to put on the live demonstration, which we have for scope of SC open system.
Next, I am going to present
Carry on, please.
you all our SCA evolution software, which is our open system. This is, you can see it in the screen. It's composed mainly by three components: a camera, which is a high-resolution camera from a German brand, connected to a microscope, which needs to fulfill different requirements, and then a computer. After, some other optional components can be added. For example, a printer, label printer, a barcode reader, a fluorescence module, and other components. But at least you need to have these three. It's also possible to check the minimum requirements that these three hardware components need to accomplish, and then maybe some components that you already have in your laboratory can accomplish these requirements, and you can use them. Also, it's important to know that this software, this system is modular, in the sense that it's not necessary to have all of the analysis modules at the same time, but you can have just one for the moment, or a couple of them. For example, motility and concentration module and morphology, and then in the future incorporate other ones. So the available analysis modules that there are, the automatic modules, are motility and concentration analysis, which will be done at the same time, morphology, DNA fragmentation, vitality, acroson reaction, and leukocytes. So this is the main information about the system. It's different from our closed system, which is the most recent one, which is called SCA Scope. You see that they are quite different, at least in the hardware type, because this is an all-in-one system which contains a motorized worm stage inside, and that we can add up to 4 slides at the same time and perform the different analyses. This SCA scope has no fluorescence, so the models that are available here are the ones that can be run in the routine of a laboratory. So these are the models that are available, and this is more automatic. But today we are going to focus on SCA evolution. So for this, I will open the software and make you a demonstration of the software. So this is SCA evolution software. This is the first screen that you can find, and this screen is allowing you to choose the user that is going to work with the system. By default, there are 3 users created that have, for the moment, different rights upon the system. For example, the administrator will be the one able to create samples, create analysis, run them, delete, change configurations. So they will be able to do everything. This will be different from the analyst, for example, that has just some right during analysis process, but after there will be some actions not allowed for the analyst. And the last one is the viewer, just able to see the already performed analysis and have the reports. It's possible to create and to customize this user window according to the people that are going to use the system in each center. And we can assign a password. Also from here, it's possible to choose the language. And then it's another important subject regarding SCA evolution. It's to understand that there are different system types and different editions. System types mean that Normally, the system that is composed by the camera, the computer, and the microscope is called a full system. That in centers in which they just have one SCA, they will have full one. But it's possible to have different combinations of the software in case that there is more than one system in one center, or there are different clinics belonging to the same group that they want to share information. Maybe if in some clinic they just capture, they just receive the images and the samples, sorry, and then they won't do the analysis right there. It's possible to have a capture system. This capture system will include also the camera and the computer, but it will be cheaper because the analysis won't be done here, but in the full system that will be linked with this capture system. So it's just a capture station. Also, there is another possibility, which is the editor. The editor system won't require a camera because in this case the analysis will be open, and then manual modifications could be done, checking the analysis, but just already analyzed images. And finally, the viewer. The viewer can be installed in any computer in order to check the already performed analysis without any modification. It won't be possible to perform any modification, but just review the different analyses. For example, the doctor could do this and print the report. On the other hand, there are different editions about the SCA evolution software. In this case, I am going to show you human, but it's possible to have other ones. For example, for veterinary is for all of the species except rat and mouse. Toxicology edition is specifically for rat and mouse samples. Production, it's very focused on production centers, only working with one species and wanting to have just the main information about the analysis. And research, with which we can analyze both human and all kinds of animal samples. So for the moment, I will log in as administrator. And we arrive to the main screen, which is the new sample creation screen. So here what you can see. In this case, the first thing normal to do when we receive a sample is to create a new sample into the software. Here there are two possibilities. Maybe we need to create all of the samples from here, from the software. So at the beginning of the day, all of the samples can be created, and then every time that we want to analyze each of them, just select the sample and then begin the analysis. But there is another possibility. Maybe it's possible to connect the software with the laboratory information system. In this case, all of the samples will appear here in the right part of the screen, and then it will be required just to select the sample that we want to analyze and start the analysis. So there are these two different possibilities. In my case, I will show you how to create a new sample and a new patient. You see here that there is a reference by default. This is the same reference that was created with the last sample with 1 unit more. This is to avoid sample duplicates. The reference of the sample needs to be unique for each sample. It's not possible to repeat. So this number will be automatically always different, but it's possible also to change it. So important, the sample reference is linked to the sample that we are about to analyze. In this case, I can introduce, for example, today's date, and then just like this, and it can be sample number 1, for example. So once that we have completed the reference of the sample, then we need to link the clinical history number for the patient. In this case, we can click here on search patient, and then this window will be open with the patients that are already in the database. So in this case, we can select a patient because maybe 3 months ago we already analyzed a sample from this patient and now we have again another sample, so at the end it will be possible to compare these two samples. If not, we can just create a new patient. And when creating a new patient, it's possible to add different information, but at least in order to save the patient, we need to fulfill two informations, which is the clinical history number of the patient. That can be a number that each center assigns. For example, I will go again with today's date and the first name. For example, George. Other informations, for example, the last name, the email, the birth date, the language of the report, and other optionals can be also fulfilled by the patient, for the patient. So here the purpose is to have all of the information about the patient collected, because it will be possible to transfer this information to the final report. So for example, if you want to add the telephone number of the patient or the wife's name or previous diseases, it's possible by double-clicking to change this word. And for example, phone, change this and also change this, and this will be changed for all of the patients in order to have homogeneous information. And for example, previous diseases. And then this could be fulfilled with information that you want. So once that we have finished, we can validate. We have our patient here and we will select it. So now that we have assigned to the sample a reference number and a patient, we move on to the macroscopic and microscopic data for the sample. What we find here are a set of parameters. This set of parameters are defined by a WHO manual and are very important to take into account for after interpreting the results of the different semen assessments, and then taking everything into consideration, arrive to the final diagnostic. So, for example, we have here when the sample was delivered, when the analysis was begun, the collection date, the volume, very important, the volume of the sample, because the concentration will be calculated in million per milliliter, but also in total million per sample. The pH, also the temperature at which we are working, the abstinence days, and then a set of other parameters, for example, if they're having collection difficulties, if the liquefaction is complete or not, the sample is viscous, if there are aggregations. So at the moment when we create the sample, maybe it's not possible to know if there are aggregations or agglutinations. But we can always come back to this screen once we have run the motility and concentration analysis, and then just complete the information that was missing at the first moment. And also, you see here that there are 14 optional fields that can be used to add additional information, this time about the sample. So you see that I have already changed some of these parameters. The technique is the same. If we double click here, or the same, we click right, we can change the title, we can change the word, and then add other information that can be either qualitative. For example, if we change this and we want to introduce the color of the sample, we can here introduce white, for example. So this would be qualitative information. Or quantitative. If, for example, you perform additional manual tests that are not automatic, in this case it's possible to perform them in the microscope and then add the results here. So this can be done, for example, if you perform the ROS test, the oxidative stress test, or the antibody anti-sperm test, for example, EGA, you can directly introduce here the the result of this test by writing, for example, this number. Or if you double click, a manual counter will be open, and you will be able to use the 0 and the dot in the keyboard to count positive sperms and negative sperms concerning the IgA test, and then link that in this field we are assessing IgA. So if you count different cells in the microscope, you can use the keyboard of the computer as a counter and then save, and you will have here the results and the units. So this can be used for additional tests, manual tests, and then all of this will be transferred in the report. So once that we have completed the macroscopic information, we can move on to microscopic data, microscopic analysis. So here we open programs, and then we begin with the first analysis recommended to do during the first hour after ejaculation, which is motility and concentration that will be performed at the same time. So I click here in motility, and then as you see in each module, analysis module, it's possible to create different configurations in case that we analyze samples at different conditions. For example, parameters for a fresh sample, fresh ejaculate, won't be the same that for capacitated sperms, or for example, if we perform motility in underfluorescence. So it's possible to adapt just to click and begin the analysis. Also, here, if you have introduced the volume, here the volume will be transferred, and it's possible to introduce a dilution factor. In this case, it's important to take into account that the software will analyze properly samples between 1 and 100 million per milliliter of sperms. With samples that have a concentration under 1 million per milliliter, maybe there are some centers that directly do the analysis manually. They concentrate the sample and follow the guidelines of WHO. And for samples having a concentration higher than 100 million per milliliter, in this case, it's recommended to dilute the sample. And this is a message that you will obtain through the software. When you get a higher concentration, higher than 100 million per milliliter, the software will show a message advising to dilute. And to dilute, you can use seminal plasma or a saline solution or other media that you have available. In this case, the next time we will dilute a fraction, an aliquot of the sample, just to do the motility and concentration analysis. So in this case, you can dilute the double, or well, a half or a third of the sample. And the way to introduce the dilution factor inside this module is to think about it as a ratio. Normally 1,0 means one part of sample and any part of diluent. But 1,1 means that we have mixed one part of sample and one part of diluent. For example, that we have mixed 10 microliters of sample and 10 microliters of seminal plasma. And the same thing. If we put 1,5, this could mean that we have used 10 microliters of sample and 50 microliters of diluent. So this would work as a proportion. Having said this, we can also keep talking about motility and concentration analysis. In this case, here you have the configuration, and we will work in positive phase contrast and with a 10 times objective. And here in the configuration, for example, you have the parameters that are followed during this analysis, which are the same, the ones given by WHO manual, but it's possible to change them. So if we click here, analyze, we will need to load our counting chamber, put it in the microscope, focus, adjust the light, and then begin to capture. So for capturing, we need to keep clicking analyze. So we can click analyze or the space bar in the keyboard, and here we can see the progression of the fields and the number of fields and the number of sperms that we have analyzed. In this case, it's recommended to analyze at least 3 fields or 200 sperms. But of course, as much sperms are counted, the better will be the analysis and the more accurate will be the analysis. In this case, we have analyzed 360 sperms in 5 fields, and the software asks us if we want to see the final results. So we can click on yes, and we will arrive to the screen of the results. So here, first of all, before checking the results, what we can do is to check the homogeneity of the sample. If we go to comparison tab, we can test, we can check the color of the videos that have been captured. In this case, there are 2 of them which are red, meaning that they are heterogeneous. They have a variation superior to 20% in comparison with the average of the other fields. In this case, we could open the field to check what has happened, if there is some bubble or there is an aggregation or some kind of problem. And if you see a big problem in the field, with the right click, we can just delete this field. If not, if for example the sample is viscous, maybe we can assume that it will be heterogeneous and we can keep working like this. And also, before going deeper into the results, we can, with right click, view the field. And these are the kind of videos, the 1-second videos that will be recorded during motility and concentration analysis. Here you can see the type of image that we expect to find when using positive phase contrast. These are the sperms with white heads, the background is gray, and then there are some particles in the background that may be artifacts and that we are not interested in black. So just the sperm heads will be analyzed. And you see that there are different colors for each sperm. This is because now we are seeing the WHO 5th edition classification into 3 categories: progressive, the red ones, non-progressive, and immotile. It's also possible to select WHO 4th edition into 4 categories: rapid progressive, type A, type B, C, or D. So you can check this. This classification will be the same as in WHO 6th edition. And in fact, nowadays we are in process of adapting the software to the new version of the manual. So we will come back to this classification. Also, it's possible from this screen to count the concentration of round cells. Round cells are all these kind of cells that are not sperms and can be found in semen sample. That can be, for example, germinal cells or leukocytes. So these are the kind of round cells. For example, this one or this one. In this case, for the moment, we cannot distinguish the origin. But after, if the concentration of round cells is higher than 1 million per milliliter, then WHO recommends to perform leukocytes analysis in order to discard a possible infection of the sample. Other thing important is that we can just double click on one sperm in order to see the individual information about this sperm, all of the velocities. These are the curvilinear velocity, the average velocity, straight line. So these are individual information about this sperm, and you see that the video, the analysis are automatic, but it's possible also to perform manual modifications. Here in the left part of the screen there is a toolbox that can be used to add or delete some sperms. So if you want to improve your analysis, it's possible. If you see that some sperm has not been analyzed or due to reasons of focus or contrast, for example this one, here in the bottom, this would be an immotile. So you can click here in Add Sperm manually and add a type D. And every time that you do modifications, the results are recalculated automatically. So you can do some modifications or even delete something that it's not an sperm by clicking here in the cross and clicking here in the screen, for example. Also, it's possible with the right click to select areas in order to delete the analysis. This can be used, for example, if you are facing to an aggregation or other type of problems during analysis. You can definitely try to remove one big area or undo your changes. So going back again to the results, we can check different results. On the one hand, the concentration in million per milliliter and in million per sample. Also, the different percentages of motility and progressivity. And here we have the 4 categories for the motility: type A, B, C, or D. And here in the left part of the screen, for the moment, we have the diagnostic of our sample. If we want to have more information, we can go here to results 2 and check some functional tests that can be also calculated during the motility and concentration module, which is, for example, mucus penetration. Here, mucus penetration is a label, it's a subpopulation of sperms that the software can calculate an approximation of how many sperms from the sample in physiological conditions could be able to cross cervical mucus in female tract. These sperms will be selected by the way that they swim, the speed, and if they are moving forward or not, if they are progressive or not. In this case, we will have the approximation, the calculation, and we will have here the results of, for example, in this case, 6 million, almost 7 million of sperms in this sample are correct, would be able in physiological conditions to cross cervical mucus. This can also help us to choose the technique for the insemination. Also, hyperactivation can be studied. In this case, in a fresh sample, it doesn't make sense to test hyperactivation because normally it's 0% in an ejaculate, but in a post-treated sample or a sample that we have incubated with an hyperactivation media, it's possible to check the percentage of hyperactivation based on the high speed that the sperms have when they are hyperactivated and also on the star-like movement that they adopt. So in this case, it would be possible also to test the functionality of the sperms, if they are able to be hyperactivated or not. And finally, also the concentration of round cells. In this case, it's important this value, the concentration in million per milliliter of round cells. Here it's almost 1, which could be the cutoff to continue studying the sample to differentiate the origin of the problem. Next, there are some parameters linked to the kinematic of the sample. This is more focused on the research, but you have the average of the velocity, the curvilinear velocity, the average velocity, the laterality of the head, and other values. And the complete list of sperm that have been captured during this analysis. This table can be exported to an Excel file by obtaining the report here, and when clicking here in spermatozoa, this table will be automatically exported in an Excel file. If not, it's also possible to create our own table, selecting just the columns and the parameters that we are interested in. In this case, it will be also possible to create, to export this data to an Excel file. So this is concerning the motility and concentration analysis. So now we will save and close. Every time that we close an analysis, it will be automatically linked to our sample, and all of the videos will be automatically included. and saved in the computer. Moving on to the next analysis, which is chronologically vitality. Vitality is also a time-dependent analysis, and it's recommended by WHO to perform it during the first hour after ejaculation. So, vitality is not mandatory for all of the samples, just for those samples with low motility. Why? Because we could be interested in knowing if the immotile sperms that we find in our sample are alive or are dead. In this case, we could be facing two very different problems, if they are dead or if they are just immotile. Maybe because there is some problem during the motility acquisition, during spermatogenesis, or if they are dead, they may be linked with other problems. For vitality, the kit that we suggest is a solution which is called Brightbit, which is a solution based on eosin nigrosin recommended by WHO. This analysis can also be done under fluorescence, but normally with the Brightbit it's easier to do it with using bright field in the microscope. So all of the models, you will see that they work the same. So in this case, we can go again to analyze and capture some images. In this case, at least 200 sperms would be recommended. So I will capture some of the images. Of course, here the technician needs to prepare the sample and then put the sample in the microscope and focus it. So once that we have captured some images, we can go again to the results and it will be the same thing. If we view this field, you see that here there are some sperms with a green square or with a red square. These are classified as alive and the other ones are classified as dead. So this is the difference. These colors can be changed. Sometimes alive sperms can be in blue and dead sperms in red also. And here with this solution, what we are testing for is the membrane integrity. So eosin solution, eosin will just enter to the head of those sperms that are dead, and eosin won't be able to enter to the heads of alive sperm. So for this, you can test for the difference. Of course, also we have here the toolbox that can be used to do manual modifications. And finally, find the results of alive and dead sperms. So I will also save and close. We move to the next analysis, which is morphology. For morphology, different stainings are compatible with the software. DivQuick, Papa Nicolau, and others that I can show you. DivQuick, Papa Nicolau, Shore, CellView, Sperm Blue, Sperm Stain, and other ones. And also it's possible to choose different criteria for morphology analysis. We can choose the WHO, Kruger, or David Modifier, or even we can create our own normal sperm values, ranges, and we can customize this screen. But now we are going to leave WHO and then begin the analysis. In this case, the technician would need to prepare the morphology smears and then stain them, and then just begin to analyze. The morphology analysis can be done, as we are using a high resolution camera, can be done with a 60 times objective at bright field. For vitality, I haven't said that, we can use 10 or 20 times objective, but for morphology 60 times. And we recommend to use mounting media, like for example EUKIT, because even though we are not working at 100 times objective, still we are working at high magnification, and then always the focus and the coloration are easy to do with a mounted slide. So I can capture some sperms and then go back to the results. So for morphology, you see that some sperms are classified as abnormal and others as normal. And if we double click on one sperm, we can see why this sperm has been classified as abnormal, because of these parameters, and then also the classification that the software has given to this sperm. So it's possible also to do manual modifications. And you see that here the acrosome, it's yellow, the post-acrosome, it's blue, there is also the midpiece and the tail. So all of these structures are taken into account during the analysis. And this is the kind of coloration that sperm blue staining provides. You see that there is a good contrast between the acrosome and the post-acrosome, and other structures can be also studied. To facilitate things, if manual modifications are required, there is this window, which is edit window, in which we obtain the complete list of sperms analyzed during morphology analysis, and it's possible to filter by abnormality index, having at the top of the list the most normal sperms and at the bottom the most abnormal ones, with more parameters abnormal. In this case, we can from here modify the manual modifications, results being recalculated, and also delete some sperms, or delete multiple ones if they are not sperms. Regarding the results, as you can see, the results are expressed like the following: percentage of normal sperms, percentage of abnormal sperms, and then all of the abnormalities counted in general. So in this case, there are 60% of sperms with head defects, 40% with midpiece defects, also tail defects, and cytoplasmic droplets. But if we want to know more, we can go also to this screen sort, in which 3 indexes are calculated. The 3 indexes recommended by WHO, which is the teratospermy index, multiple abnormalities index, and deformity index. So these can, of course, be taken into account, not just to stay with the percentage of normal sperms, but also to know more about the abnormal fraction of the sample. In morphology, we have more detailed the abnormalities of all of the sperms. This is according to WHO, but again, the criteria can be selected, but can be chosen. And the main values for morphometry. In this case, this can be used more in research, not that in routinary analysis, but in research this can be used to publish some studies and compare different types of samples. And also we have the advanced option here. So I will save and close again. We can move on to the next analysis module, which is DNA fragmentation. In this case, DNA fragmentation is studied thanks to the technique called sperm chromatin dispersion technique, or also the halo test. In this case, we are going to incubate our sample with different solutions that will produce that intact, correct sperms will generate a big halo around the heads, and in contrast, damaged sperms will not generate any halo. And then we will stain the slides in order to appreciate the halos. So if we go here to analyze, we are going to do this analysis also with 10 or 20 times objective and in bright field. And the software will take into account the halo area to classify each sperm on fragmented or non-fragmented. You see, this is, for example, a fragmented small halo sperm, but all of these are non-fragmented. You see, around the heads. Fragmentation analysis, it's not a basic routinary analysis, but when maybe the other sperm assessments, the other sperm parameters are normal, in some cases the problem could be linked with DNA fragmentation, not just in problems related with fertilization, but also with embryo development. So this is a more advanced analysis, but that in the last years it has gained some importance, and more clinics are running this test lately. So this is a kind of image that you can get. And when clicking on one sperm, we can see the individual information about the halo area and the classification. So we can take a look at the percentage of not fragmented and fragmented results, sperms, and also the number by the halo. For example, big halo or medium halo, small, without, or degraded. So I will also save and close. We move on to the next analysis, which is Ackerson reaction. For the Ackerson reaction, we need fluorescence. This is an additional module, and because this can be only run with the use of fluorescence, and two fluorochromes, which are Hodge and PNA. PNA is a fluorescence dye that is specific for the Ackerson. So we will see two types of sperms here. Some sperms with intact Ackerson, which will have this kind of coloration, green thanks to the PNA, and blue for the rest of the Hodge, and other ones with exploded Ackerson, reacted Ackerson, just blue. So we will study a control situation, the ejaculate, capacitated, and then we can save these results and then do again the Ackerson reaction analysis, but with a sample that normally is incubated with calcium ionophore in order to force Ackerson reaction. In this case, this is a treated sample, this is a sample of a study, and we can capture other images, for example, these ones. And in this case, we normally expect to find more sperms without Ackerson because we have incubated them with calcium ionophore, which forces this Ackerson reaction. So if we save the images, then if we go here to Ackerson, we can check the difference between the two conditions, control condition and the reacted, the sample incubated with calcium. And this difference is expected to be higher than 15%. If not, we can then test for the different origin problems. And finally, we are going to talk about the leukocytes analysis. Leukocytes analysis is also not mandatory, and it's recommended to be performed during also the first hour after ejaculation to discard the presence of leukocytes in the sample. So in this case, this test is done at bright field and with the objective of 40 times. And the sample needs to be stained with a substrate that it's prepared, a fresh substrate, and then just the peroxidase positive samples, meaning the leukocytes, will be stained in dark brown. The negatives will be without color. These without color, for example, these ones will be germinal cells. They are peroxidase negative. But the leukocytes will be dark brown, will be positive. So at the end, we will have the concentration of peroxidase positive cells, leukocyte cells. And if this concentration is higher than 1 million per milliliter, then it will be not recommended to use this sample for art techniques. So we can save and close. And finally, just to mention that there is a manual counter, which is not an automatic module, but it's a manual counter that provides us with the images that we can capture, but it works the same as the other modules, but here the classification is manual. So we can run any analysis with this manual counter, but we would need to do the classification. Okay. And finally, the final step after performing a complete analysis that I have shown you is obtaining the reports. By default, there are a lot of reports that SCA Evolution provides us with. There is, for example, a summary report with the main results of the analysis. And it's easy, it's possible to export this report in PDF, in Word, or to directly print the report. Or also it's possible to obtain a complete report. containing a lot of information, also some graphics, some images, the macroscopic information about the patient, and all of this. And it's also possible to export in Excel, in Word, in PDF, or other formats. I imagine that you have seen also the validation option. It's possible to enable the validation in the cases in the hospitals in which there are some laboratory technicians, and then some biologist or someone else needs to check the analysis before sending the report, the final report, to the patient. Also to mention that here in Microoptic we count with strong technical support. During our working hours, you can also you can just ask for remote support, and we will help you by seeing your screen and taking the control of the mouse. So if any error message in the screen or any problem or doubt, it's possible to ask for remote support, and online in the distance we will help you. And the majority of the problems can be solved like this. Okay, so I hope you have enjoyed this demo, and thank you for your attention.
So thank you, Gloria, for the demonstration and for the recorded video. So we have some questions, and I request the faculty to be online. So ma'am, if you permit, we can take these questions. But these questions were from Dr. Vasan's talk. Actually, he's not here with us now.
Okay.
Okay. So we had a question from a attendee that given that AO emission spectrum is much lesser than 670 filter, you may be losing much of the red signal. How do you compensate for the loss of crucial fragmented DNA signal? And what is the software used to get the report values? Here's the question.
Okay, can you repeat please the question? Just the beginning of it.
Given that AO emission spectrum is much lesser using a 670 filter, you may be losing much of the red signal. How do you compensate for this loss of crucial fragmented DNA signal? And what is the software used to get the reports?
Okay, in the case of DNA fragmentation analysis, we have been using the bright field, not fluorescence. So in this case, in general, with this technique, sperm chromatin dispersion technique, it's like a quick screening of the fragmentation of the sample. And of course, using the complementary techniques as the COMET or the tunnel or other toluidine, other techniques available in the market, we can do a wider analysis. But with this kind of technique, it gives us a first screening of the fragmentation of the sample. I don't know if I have answered your question.
Yeah, actually, Gloria, this question was not for you. It was a question for the previous speaker who left the chat now. But thanks to you that you answered it. It was really helpful.
Okay, sorry. I thought that it was for me.
I was utilizing you because you were available with us now.
Okay. Well, I had tried to answer. I hope that. Okay.
Thank you so much. Yeah.
Okay.
Because I have to say the questions to the faculty, but we don't have faculty with us now. So, ma'am, I think we can close for now because we have completed all the lectures. And tech team, if we have any more questions, you can type in the chat box. If not,
कुछ तो question पूछ लीजिए.
Okay, ma'am, there are a few more questions. So, Dr. Jindal, I will need your help now because Dr. Vasan is not here. So, you are the senior person among us. So there was one question by Mr. Nikhil. Recent studies on developing of placenta says that a placental development is responsible by reserved viral DNA inside the genomic DNA. Is this true or false? It correlates with doesn't correlate with any of us now. Gloria, any insights on this from your side?
I am not sure. I am not sure.
No issues.
I don't know.
So regarding Gloria, regarding your lecture, so
yeah.
Related to the SCA equipment. So what, for example, if I am a pathology laboratory, and I want to opt for the QC program. Okay.
Yeah.
As per now in India, we don't have the QC program available till now. With any, not the EQUAS program is still not in the picture itself. Because for pathology, for biochemistry, there are programs for external quality assurance scheme, but for semen analysis, it is not. I think I will say it's not practically possible. But with the introduction of your internal QC program, which you provide the beads, in which you provide the pre-stained slides, I think that could be a helpful thing. And is it available in India?
Yeah, this is available in all countries. This is something that can be done once the one laboratory has the software. They can obtain the QC beads, the pre-stained slides. They come with all of the SE scope devices that are sold. Sorry, and also, for example, the micro-optic quality control that I have explained, which are this set of sessions that can be analyzed by all technicians in a laboratory. This is also, well, included. So yeah, definitely in all countries.
So we need to monitor these following points.
Interlab comparison. Like for example, if I am doing my lab, I am running in my lab, and Gaurav is lab too nearby my hospital. For example, if I am doing something and I send the same sample to Gaurav, analyze the same, then we compare a result. Is that be a feasible option if I'm not getting an external quality assurance scheme in India?
I wouldn't say that this is 100% equivalent to an external quality assurance program, because it's true that this kind of quality assurance program, EQA program, compare different interlaboratory results, and this micro-optic quality control is more to make sure that all technicians using the program in one center share the same criteria. So it's not that standard, let's say, but it could be a first approach in order to know if the software, the semen analysis software, is well used by the technicians, which it's a first step and it's very important.
Okay, so next question to you regarding surgical sperm retrieval. So you can unmute yourself now.
Okay, okay,
yeah.
Thank you.
Dr. Jindal, I have some queries. Like for example, if I'm like I'm not a very sound or a very experienced embryologist with respect to surgical retrieval. So is there any option, like when you gave me the tubules, okay, and I'm getting so many testicular cells as well as the sperm also. So is there any few tip points which you can give to a young embryologist who is dealing with surgical retrieved sperm by the urologist? Because you will be at that time, urologist or the surgeon or the gynecologist will be in the OT and I will be sitting in the lab. So some tips and tricks which can help me find good sperms in that testicular homogeneous suspension which I get.
See, there are 4 things which I follow. So I'll just tell you that.
Please, sir, please.
And what I follow are very simple. Yep. Whenever it is being sent into the theater, I would normally say, do not know. I think there are. Please keep this specimen.
Okay.
So these are 3 things I always say. So what happens is that once you've opened the testes and you are just looking at it, it's not initially, but after some time you gain an idea by the look, feel, color, and the turgidity of the tubules that these probably will have. So take them out into a different Eppendorf tube. You do not put them into every tube which you have made.
Yes.
So instead of putting them onto anything else, now I use only Eppendorf tubes. So what I do is I'll put them into a different Eppendorf tube, and I will say immediately, the guy who's taking it inside the lab, just take this and ask him to do it before the other specimens.
Oh, yes.
So if he has teased it into a different dish and he finds sperms, then I have an idea, these are the tubules, this look of the tubules I've got to look for.
Yes.
So that's a very simple thing to do. And you've just got to, if you feel these are the tubules right, you've got them put, you don't chunk them into everything else. No, put them into a different tube, send them and ask him if they are there. And if they are there, then you visually know this is what I need to pick up.
Yes.
That's what I do.
And sir, regarding the safety of pentoxyphylline and theophylline, because there are like 2 schools of thoughts in the market.
Yes.
Like, yeah, so on that.
Yes. What we do is, We use the sperm flexibility test in most of these sperms.
Okay.
So a lot of times we are sure that it's fine, it's okay. Otherwise, there's a new media which has come in. I've forgotten the name of that. That has theophylline.
Okay.
That has theophylline.
Yes, sir.
So we used to be using the pentoxyphylline earlier. For the last 2 months, we are using theophylline.
Okay.
So I really would be able to tell you better once 6 months have passed.
Okay.
Pentoxyphylline is fine. I mean, we use it, but we use it rarely. We normally buy sperm flexibility. I mean, my guys, we just come to know about it.
And sir, from some, yeah, please, ma'am, please.
Hello, sir.
Hi, how are you?
So now when everyone says, earlier it was that DNA fragmentation, if there's higher DNA fragmentation, we go for testicular sperms. But there are various studies now suggesting there's no change or no.
I never do it. I never do it.
You never do
Never,
what?
never. Do a testicular sperm unnecessarily.
So I was just about to say that, are you also very against doing invasive until and unless it is required, and not to go for invasive procedure?
No, I'll tell you. I'll tell you what I do. I'll be able to tell you after reading everything. So what we normally do is that in case we get a sperm which is normal in morphology and it looks fine, we would use an ejaculated sperm. We would not use a testicular sperm just because we need to do a testicular sperm. Now, if the patient has had a history of an abortion with a low count ejaculated sperm post ICSI, we would go in for a testicular sperm.
So, but let's say if the patient has a very high DFI, would you, if you know the previous cycles haven't worked, would you go for a testicular sperm extraction for that patient?
I'd say his wife, if she is young, I probably will not. And if she herself is of a particular age where I feel that the eggs too would be having something, I may go in for a testicular sperm. I mean, it is something which you cannot quantify. A lot of people are going into testicular sperm now. And I don't really know if it is justified. I don't know. And even literature today doesn't prove that it's a justification.
So my point was, after all these studies of DFI, DNA fragmentation and everything, but when it comes to the lab or a practical aspect of it, what can we do about it?
Yes.
You know, it's...
you know what? It's very simple. If there is a person who's come with a high DFI and you're sure you've done it properly, you'll check if the patient has leukocytospermia or the patient has any infection. If infection is present, it needs to be treated at all cost. One, you will check if the person has varicocele. Please get the varicocele operated by microsurgical technique. Third thing is put the patient on antioxidants, which is no problem. Vitamin E and vitamin C, zinc, selenium, folic acid along with coenzyme Q should be part of the armamentarium which you're giving to them. Once you've given it to them for 3 months, check, recheck the DFI. If the DFI has reduced and it's come to a reasonable level, then go ahead. I mean, that's all that you can do. There's nothing else you can do.
So how many patients are patient enough to wait for 3 months, taking the antioxidants and everything, and then coming back?
You know something, Saroj, I'll be honest with you. If the patient trusts you and you trust the patient, he will not leave, they wait.
So I let's say 90% believe in what you're saying, but these days, like I've been doing embryology for so many years, patience level of the patients is dying.
Yes. It is. What you're saying is right. But then what else?
Don't wait for those 2-3 months to, let's say, improve the oocyte quality or the sperm quality. They want instant results. यही मतलब this month यही month मेरा cycle कर दीजिए, जो होना है यही month में हो जाना है. We are not agreeing to wait for another 2-3 months. How do you handle these kind of things then?
I only tell them one thing when I'm in conversation with them. I spend a little bit of time and I love human beings. Let me be honest with you. So when I sit down with them and I'm just listening to them, talking to them, and I tell them that we're on the same side of the table. You want a baby, I want to give you a baby. And we need to work on it. And to work on it, we need 2 months. I think you want a baby. That's what you want. Believe me, these 2 months may do a difference. All of them wait. And whenever they say, मैं जल्दी में हूँ, तो मैं कहता हूँ जल्दी में तो अभी करा लो, अभी करा लो, अभी करा लो. और बच्चा नहीं होगा तो मुझसे मत कहना. I mean, it's very interesting. All the people, I'll just share something. It's a different thing I'm sharing with you. Whenever people, the simplest thing is to just listen to the patient. In a minute, you know what's his biggest pain point. His biggest pain point may be the mother-in-law. The biggest pain point may be the fact that the husband has an affair somewhere else. And he says, I'm going to marry that other woman. And the moment you tell her, कि सुनो, तुम्हारी सास को जब पोता मिलेगा कितनी खुश होगी. Believe me, she doesn't care what you're doing. She doesn't care about your equipment. She doesn't care that you're going to do an ICSI, you're going to do a PGT, you're going to do anything. Her pain is the mother-in-law.
True.
So it's just a question of sitting and listening to her for a minute and telling her to wait, please, because your husband will love you even more. Period.
Was that easy, sir?
I mean, I just had to say this because that's what you need to tell them.
I think we had a fantastic workshop. Gaurav, anything to say? Is here, is like on and off there.
Yeah.
Thank you, sir, for a wonderful talk, sir. Sunil, sir, it's always my pleasure to listen to your talks. Thank you, Gloria. That was a wonderful presentation on SEA. Yes, Gaurav?
I think we are done for the day. We all are tired. We enjoyed a lot. It's always pleasure looking at Dr. Sunil, Saroj, Vakil, and Neva people. Been a long time.
It
एक
was
physical भी होनी चाहिए.
workshop. We learned a lot today, and I'm sure all the juniors would have really, really loved whatever came their way because it was handled quite practically and interactive. So, thank you guys. Good night. I think it's
Thank
time.
you. Thank you.
Thank you.
Thank you very much. Thank you, Gloria. Thank you, Gaurav. Thank you,
Bye-bye.
Vakil. And thank you, Saroj, as always. Say, give my best wishes to your wonderful and handsome husband. Okay. Bye-bye.
Bye.
Thank you, everyone.
Bye.
Thank you,
Bye.
teammates. Thank you, tech team. Bye-bye.
Bye.
Bye bye.
चलो भाई टाटा बाय बाय. हाँ ब्रो.